we don't actually. Idea is I'm going to introduce cognitive science, right? The biophysics of the brain. And also I'm going to sort of introduce some food for thought, like what sensors we use. We usually use sensors out from the vendors and also from the companies. So per se, I'll introduce what the sensors for various brain monitoring applications are out there. And also I'll try to introduce some of the research, but again, the research papers which I would discuss would not go in depth from a sensor point of view, but I think this will definitely inculcate interest in you, and I look forward to your feedback and comments and questions. Okay. So let's get into the talk. Okay, so this is going to be the bird's eye view of my talk. So I'm going to sort of introduce to you the human brain monitoring techniques which are currently used. And also after introducing a few modalities, that is the brain monitoring techniques, we will pick one modality in the sense, one monitoring technique, which is electroencephalogram. And we will go in depth into the biophysics and the principles of the physiological basis of electroencephalogram recording. Okay, from there we will move on to look at some of the sensors which are being used to record electroencephalogram, and then I will sort of move towards a cognitive side, wherein I will introduce the cognitive paradigm P300, which will enable some evoking of the brain during an experiment for a subject, and then I will show you how exactly a brain-computer interface can be designed. Okay. So pictures actually communicate 1,000 words. So this is sort of the view of the various sensors which can be placed onto the human brain for monitoring it. Okay, and this is sort of the brain-computer interface. Bird's eye view diagram, wherein a subject is doing an experiment and he's trying to spell a letter. Let's say the letter H first, and then followed by the letter I. Okay. So this sort of gives you a bird's eye view of the talk. Let me get into the talk. So I think most of you know about a lot of things about the human brain. It's the most powerful organ of our human being, right? So some of the facts: there are nearly 100 million billion neurons present in the brain, and the storage capacity is virtually considered unlimited. And the neurons start developing as early as in pregnancy, and it develops at the rate of 250,000 per minute. So if you calculate the brain information rate which travels per hour, right? So if we keep thinking per hour, we can say that our brain is sort of traveling around 268 miles, right? And then oxygen, oxygen is very much needed for brain. So it can maximum survive for like 4 to 6 minutes. And after which it unfortunately sort of gets into the dead state, right? So moving on. Okay, so this is a functional anatomy of the brain regions. So this is actually the light pink is actually the prefrontal cortex, which is considered as the working memory of a spatial task, and it's also responsible for solving complex multitask problems. Okay, and also it's considered so the executive area of task management. So this is sort of the executive control of our whole body, and all the commands are generated from here. And then if let's say you're doing some vision related stuff, so it's actually the primary visual cortex, which is this part of the brain. Okay, and then you have the visual association areas are also depicted. So similarly for other tasks, so let's say if some hearing task is involved, so then you have your auditory association areas and also the primary auditory cortex, which is depicted over here. So roughly you can say the brain, the predominant areas of the brain are the prefrontal cortex, the motor areas, the movement related stuff, and the sensory areas and the associated areas, which act together with the primary lobes of the brain to perform any function. Okay. Okay, so now I've introduced you to brain, and I've also showed you some functional functions of the brain. So if you want to measure the function or the structure or study the structure of a brain, what are the various modalities? Modalities in the sense, the methods or the equipments or the sensors which are available out there to study a human brain. So let's quickly look at them. So you have a lot of possibilities to select various modalities. The first one being the electroencephalogram. Okay, so electroencephalogram is nothing but the summation of the postsynaptic potentials at the dendrites of a ensemble of a neurons. Okay, so this is what we are going to look in details. Hence, I've actually marked this. So we are going to select this modality and go in depth and see how we can realize a brain-computer interface using an electroencephalogram, and what are the sensors currently available in the market, and what are the sensors which are in the research. So I'll discuss a few papers where they are coming up with novel nano sensors. So that might actually be of interest to you. So apart from this, which measures the neuronal activity at the postsynaptic potentials, there's also FNIRS, which is an optodes. So you have your source and the detectors. So it's based on the optical principles. So this also has the capability of measuring the functions of your brain. So the other one is the magnetoencephalogram, which actually measures the magnetic current, which is magnetic fields which are created by the electric fields of your brain. So this is actually the fMRI. You might have seen this in all the hospitals. So this actually enables us to differentiate between the soft tissue types. So like the brain has white matter, gray matter, and cerebrospinal fluid. So if you want to study the soft tissues, predominantly you use a structural MRI. And if, let's say, you want to find the function also, so then you will go for a functional MRI. Okay, so these are some of the modalities of the which can be used to study the brain. Okay, so how do you select which type of modality, right? So I introduced four modalities to you. So two things are very important, right? One is the space or the, because you are going to measure the brain, and there's going to be a factor called space or the space dimension. And then the other one is you're going to study the person over time, right? So that is going to be time. So, and again, if you are interested, there are two things to note over here. One is the non-invasive in the sense there's not going to be any penetration of your skull and into the deep tissues of your brain. So over here in these modalities, the sensors are actually placed on to the head of the person, and these are predominantly very safe. So you can see the sensors placed on the head of a person using a cap. Similarly, in an fMRI or an MRI scans, you're just going to, the person is going to go into the machine, and there's not going to be any penetration of your skull. But if you want to get more higher signal to noise ratio, then there are various modalities which do invasive placements of your sensors or the electrodes. So those are actually called as your invasive. So that is like the local field potentials, and also the single neuron recordings are all going to be sort of penetrating your skull, and the electrodes are going to be placed onto the cortex, or let's say even deeper into the cortex. Okay, so basically the modality depends upon your need and your application. Okay, so this is what would be the invasive versus non-invasive brain imaging methods. So this is your scalp, the scalp of your head, right? And we can place electrodes on the top. This is perfectly safe and non-invasive, like you see over here. So this is the electroencephalogram electrodes and the cap which is placed over here. And this actually records the synaptic potentials, as I told you already. And then if you can also place your ECOG or the ECOG electrodes, you can see it over here. So this actually requires a clinical team. So these electrodes are placed on the brain surface. And if you want to go a bit deeper, so this is actually around over on the surface of the cortex, but you can penetrate the cortex and you can place intracortical electrodes. Again, all this requires clinical team. So this figure actually shows the non-invasiveness of the placement of the electrodes versus the invasive nature of the placement of the electrodes to monitor the brain activity. Okay. Yeah, so this is sort of the summary of the brain imaging methods. So if you this is electroencephalography, magnetoencephalography, and fMRI and FNIRS. So the type of signals which is recorded is neuroelectric for electroencephalography, and also for the ECOG, it's neuroelectric. And if you look at the functional fMRI and the FNIRS, it's the changes in the blood flow. So over here, changes in blood flow is being measured during a task or during any given, paradigm of the person is actually making. So if you want to decipher what exactly the person is thinking or doing during the task, you need to make use of the changes in the blood flow, which is monitored by the FNIRS and the fMRIs. Okay. And then the temporal resolution would be over time. So if, let's say, you want to monitor a patient who is having epilepsy, so you would like to get the signals over time, right? So temporal dimension over here would be the signals which are being recorded over time. So electroencephalogram has a reasonable time resolution. That's why you have 3 pluses. But unfortunately, we do not know where exactly the electroencephalogram or the recording of the summation of the potentials exact point is spatial location is not possible, because it records from an ensemble of neurons, ensemble or a bunch of neurons. Okay. And again, there's no it actually there's no depth penetration like I showed over here. So it's recording over here. So it's going to be a bunch of neurons which are present in this part, right, which are going to get recorded. So we will look at that in detail as I explained to you. And then the brain coverage and invasiveness, it's not an invasive one, right? So it's a non-invasive technique. So you have a high non-invasiveness over here. Okay. So similarly, you can look at the fMRI. fMRI is actually, as I said, it refers to the monitoring of the changes in the blood oxygenation levels. And the spatial resolution is pretty high, but the temporal resolution is slightly impaired when compared to electroencephalograms. And the depth of the analysis is pretty much more higher in the fMRI than in your electroencephalogram. So again, the brain coverage is high in fMRI when compared to the brain coverage in the electroencephalogram. And of course, if you're getting an fMRI, so you have to be inside that fMRI machine. As you've seen when you visited hospitals, it's a huge machine and it's not possible to realize mobile applications. So electroencephalogram would be a better choice if you want to have a mobile application. Let's say monitor a person who is having epilepsy. You want to get his signals, but you still want the person to go around, then you could probably go for an electroencephalogram based recording. Okay. So these are some of the parameters or some of the features you consider when you want to image a brain. Okay. Okay, so I'm going to show this slide quite a few times so that I touch upon your brain and you know where exactly we are. Okay, so this is actually the bird's eye view I showed you before. So currently I've introduced you to the brain imaging techniques. So now I've selected electroencephalogram, EEG, so which we are going to go in detail. So we are going to go from a single neuron, multiple neurons, Hodgkin-Huxley model, and then how exactly the biophysics of EEG happens for the recording to be happening on the scalp of the person. Okay, and then we will look at the sensors which are commercially available as of today in the market, and also some of the sensors which are still in research, and there could be a possibility for them to come out in the future. So once I introduce these two, then I will show you how to realize a brain-computer interfaces wherein you can sort of spell a letter using just your brain waves. Okay, so this is again the picture wherein you have the EEG being recorded, some pre-processing and classification, and some based on that you sort of detect what letter this person is focusing on, and you display it. Okay. Okay, so let's quickly get into the electric fields or in the human brain or electroencephalogram. So I think this is going to be the sort of the flow, single neuron, multiple neurons, and then physiological basis. So I think most of us know this from school biology. So I'm just going to go through this quickly. So neurons are actually the specialized cells for information transfer, and they are responsible for firing of action potentials. So you have your dendrites, which receives signals from neighboring neurons. Axons transmit the signals. Soma has nucleus, and where the processing actually happens. And then the myelin sheath also enables you to speed up the signal along the axon. Okay. So types of neuron, there are various types of neuron: unipolar, bipolar, pseudo unipolar, multipolar. So it's basically depends upon the number of processes which are actually originating from your soma regions of your neuron. Okay. And the most commonly occurring one is your multipolar neuron throughout the human body. Okay. Okay, so the neuronal cell membrane. So I think you would have all heard about the cell membranes, right? So the neuronal cell membrane can be actually is made up of your lipids. So lipids are nothing but shown over here, which has a hydrophilic head and a hydrophobic tail. So this is your bilayer cell membrane. So this bilayer cell membrane is shown over here, which is made up of your lipids, which actually has a hydrophilic head and a hydrophobic tail. So you can see that this lipid molecules can be organized into a bilayer or even in this form of a structure. So the basic function of your biological membrane is it protects the content of the cell. It enables the passage of critical nutrients, and it must be flexible. Okay. So this is the cell membrane. So this is the early fluid mosaic model, and this was the later, heterogeneity was accepted. Cholesterol and phospholipids with different chemicals forming subdomains were also sort of accepted in the later versions of your cell membrane models. Okay. Okay, so I told you the cell membrane. So we have your lipids. So this is the outside of your cell membrane, and this is the inside of your cell. Okay, so these are actually the proteins. These are the proteins across your cell membrane, and there are a lot of ions across your cell membranes. So so there are two types of transporters. One is the ion transporters, which is shown over here. So these are membrane proteins which select one type of ion. So as all of you know, there are ions outside of your cell membrane and inside of your cell membrane, right? So ion transporters are membrane proteins that select one ion type and move them either inside to outside or outside to inside. So this is the ion transporters, whereas ion channels actually select the specific type of ions, and they move the ions from the outside to the inside of your cell membrane. Okay, so let me just give you a brief idea about resting membrane potential. Okay, so what is resting membrane potential? So resting membrane potential, let's say a person is not doing any work. Okay, so he's just relaxing and he's just sitting, and let's say for let's say 4-5 hours he's sitting and not doing any work, just relaxed state. So in that situation, the amount of energy or the consumed, or the energy which is used by that person is roughly not going to be that much when he is, let's say, playing football, or let's say he's doing some complex math calculations. Okay, so you can compare this resting membrane potential to something when a person is not doing anything. He's very much relaxed and not doing any work. He's just allowing his mind to wander, but he's not doing anything concrete and creative to spend energies. Okay, so let's consider a single hypothetical neuron. So we have the neuron actually boxed into a sort of a over here, and then you're connecting it to a voltmeter. So this part is the inside of your cell, and this is the outside of your cell, and this is your semi-permeable membrane. Okay, so what I'm actually doing is I'm converting this entire thing into a hypothetical boxed sort of arrangement, wherein you have the inside of the cell depicted here, and it's sort of connected to a voltmeter. So let's say if the inside of the cell is roughly having 1 millimolar of KCl, and the outside of also has 1 millimolar of KCl. So there are equal concentrations, and the voltmeter is sort of going to show a voltage of around V equal to 0. But that is actually not the case, right? So, however, in reality, there's going to be a just to get you started. I showed you this, but in the human beings, and also let's say other squid neurons, the measured ion concentrations are going to vary between the inside and the outside of the cell. So you can see in the mammalian ones, the inside of the cell, the potassium has a higher intracellular concentration when compared to your extracellular, right? And the extracellular concentrations of sodium chloride are higher in the human mammalian neuron when compared to the intracellular neurons, okay? Intracellular regions, okay? So now what exactly would be the resting potential? So you have a case where the inside of the cell has, like I showed you over here, right? The inside of the cell has the higher concentrations over here for potassium, right? So we can see that over here, the inside of the cell, the 10 millimolars of KCl. So there's some higher concentration of potassium over here when compared to your Cl minus. Okay. So because of that, what's going to happen is some of the K plus ions, the orange ones, so semi-permeable membrane is permeable and it allows some ions, especially the K plus ions, to move from the inside of the cell to the outside of the cell. Okay. So because of that movement, what happens is there's going to be an electric field which is going to be created. So because of this movement, there's going to be an electric field which is sort of created. So that electric field is depicted over here. It is shown as the positive field at the outside of the membrane and a negative field to the inside of your membrane. Okay. I hope all of you are able to hear and I'm still audible.
Yes, sir.
Okay. Yeah, just making sure, just confirming it. Yeah, thank you. Okay, so you can see that there is a electric field which is sort of created at the semi-permeable membrane. So because of that electric field, so some ions, again the positive K plus ions from the outside of your cell membrane goes into the inside of the cell membrane till there is going to be so till the diffusion flow, so till the electrophoretic force sort of equals the diffusion force. So this is your diffusion force, which creates the electric field. And because of the electric field, there are movements of your potassium ions to the inside. So so there's going to be a potential at which the diffusion force equals to your electrophoretic force, and that actually is called as the resting potential. So the resting potential is usually minus 58 volts in squid neurons. Squid is a type of fish. And for human neurons, it's sort of minus 65 millivolts around. Okay. So each type of neuron will have a different type of, different value of your resting potentials. So again, that actually can be calculated using your Nernst equation if you have only one type of ions, and Goldman's equation if you have multiple ions between the inside and outside of that particular cell. Okay. So again, as you know, the cell, each cell would have different types of ions depending upon the function, and also depending upon the organisms. Okay. So what are graded potentials? So now we saw that the resting potential is around minus 65 millivolts, right? So let's consider a hypothetical neuron, one single neuron. So this is your dendrites, this is your soma, and the green part is your axon, okay? And this is sort of going to be a resting potential. So you're going to have synapses. I think most of you would have heard about synapses. Synapses is where the information transfer between one neuron to another neuron happens. So let's say this is one synapse. And you can see another synapse over here also. And you can see that the inside of the neuronal membrane is minus 65 millivolts and the outside is actually at 0. So you can see this is the resting potential of this neuron. So what happens exactly over here is when there are multiple synapses happening, right? So this is actually the graded potentials, which is nothing but the changes in the membrane potential of the neuron. You can see that there is going to be a change in the membrane potential. So if the membrane potential changes from minus 65 towards, let's say, minus 50, then it's called depolarization. And if it becomes more negative, it's called as the hyperpolarization. Okay. So let's see the movement of ions which actually contribute to this. So you can see this, if let's say entry of Ca plus, right? If you can see the Ca plus ions entering through the membrane proteins. So if this enters, then it contributes to the depolarization. And also similarly, the entry of Na plus. So Na is entering to this membrane protein. It's going to depolarize this membrane because it's going to go towards the positive voltage. But let's say the exit of K plus. So you have K plus ions inside your neuronal membrane, and if it's exiting, so it's going to become more hyperpolarized. That is, the membrane is going to become more negative. An entry of Cl also could happen. So that means it is going to become more hyperpolarized. So this is just one scenario. So depending, that at each neuron and each synapse, in between, there are going to be bunches of neurons, and lots of such scenarios are happening. This is a very, very simple case where I'm actually showing you the depolarization and hyperpolarization. But let's say if you go in depth at a single neuron level and you try to study the synapses, there's going to be bunch of neurons with a bunch of connections, maybe the axodendrinic synapse, the axodendric synapse, various types of synapse happening in a small region. And there's going to be a lot of depolarizations and hyperpolarizations, which are going to happen in, let's say, a bunch of neurons you want to study. Okay. So what exactly would be the action potential then? So let's say we consider three synapses, right? So this is actually the synapses which are happening pretty close at this part of your dendrites of this neuron. So this is one neuron, and there are three other neurons which are connecting at this 1, 2, and 3. Okay, so they are connecting over here. They are nearby, but however, they are actually connecting at the same time. Okay, so what's exactly happening is temporospatial summations are happening, and as soon as that happens, it's going to cross this threshold potential. So once this threshold potential is crossed, then there's going to be a generation of your action potential inside your axon regions. Okay. So that's what is shown over here. You can see a single cell, and you're simulating the hyperpolarizations and depolarizations using a battery and a current source, right? And you're measuring the action potential or using a voltmeter. So when you have hyperpolarizing current, so you see there's hyperpolarizing current, but there is no action potential generated because the membranes are becoming more hyperpolarized, becoming more negative, and it's not going to cross your threshold potential. But when you sort of give a positive, when you give a depolarization current, right, you see that the membrane potential actually sort of becomes more depolarized. So because of that depolarization, when a summation of multiple depolarizations happen, it's going to cross the threshold, and then there's going to be a generation of an action potential. Okay, so you can see over here that the depolarizing current actually creates the action potential, and the hyperpolarization current doesn't cause any action potential. Okay, and again, to just to make it clear, all this depolarization and hyperpolarization happens because of the movement of your ions across the inside to the outside of your cell, or from the outside of the cell to the inside of the cell. So the various ions which we discussed, K plus, Na plus, Ca2 minus, Cl minus. Okay. So this is roughly the generation of a action potential when you consider at a neuronal level. Okay. So I hope I'm clear in these concepts. So going forward, action potential, there are many types of neurons across various organisms, and the action potentials are not going to be same. So they are going to vary depending upon the neuron, the function of the neuron, where exactly it's occurring, and also which organism it is actually present. Okay, so this shows the action potentials for that it has many shapes. Okay, so I just touch upon Hodgkin-Huxley model. So I think a lot of you might be knowing. So this is some seminal work which was done quite some time back. And the basic takeaway from this work is that electrical circuits can be used to model the ion flow across the neuronal membrane. So we discussed the resting potential, action potential, and graded potential. So all that can be modeled using the electrical circuits, and that was the basic or the contribution for Hodgkin-Huxley in their work. Okay, so you can see over here. So let's say if you have the inside of the cell and the outside of the cell, and these are the ions present. So this is your semi-permeable membrane. So the semi-permeable membrane is actually represented by your capacitor. Okay, and each of the channel can be represented using one resistor and one current source. Okay, so these are your voltage and time dependent ion channels. So you have your channel. We already discussed the channels, ion transporters. So all these can be represented using your resistor. So let's say you have your to represent the Na plus channel, right? So you have your variable RNA and you have a nurse potential corresponding to the the battery. This battery source corresponds to your nurse potential of your sodium ion. Okay, so some ion channels push the membrane potentials positive. Other ion channels push the membrane potential negative. Together, these channels give the neuron machinery flexible to control the membrane voltage. So there is a possibility to derive the circuitry of your neuronal membrane, which we just discussed using capacitors and resistors and voltage sources. So this sort of enables us to have a control over in simulations and try to see what exactly is happening in the various types of neurons rather than depending upon the neurons themselves. Okay. So these are some of the things which I discussed. So semi-permeable membrane acts as a capacitor. Each channel is represented or characterized by a resistor. Nurse potential generated by differences in ion is represented by battery. Specific batteries, since nurse potential is different for each ion. If input current is injected into the neuronal cell, it charges the capacitor or leaks through the channels, and the modeling is possible using a set of equations. So I'll not go too much into that. I'll just introduce the Hodgkin-Huxley model to show that electrical components can be used to study the neuronal transmission processes. Okay. So now I think just to wrap it up, we looked at single neuron so far. So whatever we looked at so far was on single neuron, the resting potential, the graded potential, action potential. So now let's look at, let's say, two neurons. Okay. So again, this is very simple case of one neuron and two neurons. But in reality, if you're going to record any signal, it's going to be a bunch or an ensemble of neurons. So you can just imagine how complex that recording is, and you cannot definitely tell that the recording obtained is from this neuron or that neuron. You can just say that it's from this bunch or regions of the brain. Since the electrode or the sensor is placed at this point, the recording is going to be those set of ensemble neurons. Okay. So just to show you the concept of neurotransmitters. So this is one single neuron, and this is the axon terminal endings, and this is the another neuron. You can see this one. So this is going to be another neuron. So chemicals known as neurotransmitters, you can see these are bound in membrane bound vesicles. So these can be found in the axon terminals of the, let's say, neuron number 1. So they actually are released when Ca2 plus enters, and these neurotransmitters can be excitatory or inhibitory. That is, excitatory in the sense they can fire up this second neuron, or they can also inhibit the transfer of information from this neuron to from neuron 1 to neuron 2. Okay, so that's what, and that depends upon the neurotransmitters. So if it's a glutamate, then this will actually excite or fire this neuron. But if it's, let's say, a GABA, which is an inhibitory, then this neuron is actually not fired and the information transfer doesn't happen. Okay. So again, there are various types of synapses. So there is the axodendritic synapse, which happens between the axon of the neuron number 1 with the dendrite of the neuron number 2, and axon of axosomatic synapse, axon of the first neuron with the soma of the second neuron, axonic synapse, wherein the neuron of the axon of the neuron 1 actually synapses with the axon of the neuron 2. Okay. So now I've introduced you to the happenings at a single neuronal level and also between neurons. So now let's try to understand what exactly is electroencephalogram. That is the recording of the electrical signals at the scalp of a human being. Okay, so we saw that we are going to place the electrodes or the sensors over here, right? And let's say we want to measure it's going to measure a signal around this part of your brain. Okay, so maybe this part of the brain it's going to measure. So you can see that this is the synapses. So this is the dendrites and the synaptic view, and these are the potentials which are created across the depth of your brain or the head, right? And you can see that the signal, the time domain signal is recorded for 4 seconds. And if you take a frequency plot or the frequency spectrum of it, you're going to get these frequencies. Okay, so this actually gives one viewpoint of the recording. Let me show you the other viewpoint. So if you have this electrode over here, so the gray matter actually has the neurons, the neurons about which we spoke. The neurons again have the ions inside and outside of the cell, and they have synapses. So it's quite a complex environment in this region of the gray matter of the brain. Okay, so this is actually the sulcus regions of the brain. So the sulcus and gyrus of the brains, which are sort of the ups and downs, right? So this is the sulcus regions of the brain where the gray matter is present, and in those gray matters the neurons are present, and each of the neuron will have ions inside and outside, and then there's going to be synapses and a lot of stuff happening. So what exactly is being measured at the electrode is nothing but summation of the excitatory and inhibitory postsynaptic potentials at the dendrites from the ensemble of neurons. So that is what is being recorded in an electroencephalogram recording. So the electric potentials actually generated in the gray matter region of the brain can actually be modeled as a dipole. So the radial dipole actually contributes more, and the tangential dipole actually cancels out over here. So these radial dipoles contribute more than radial dipoles which are near to the electrode contribute more than the radial dipoles which are a bit far. Okay, so this roughly shows you that what exactly is being recorded over here. So the signal which is recorded over here is shown over here. It is a summation of the lot of potentials of the neurons of the brain in that region. Okay. Okay, so these are some subfields. So, I mean, before I show you the subfields, let me show. So you recorded your signal. So from the signal, let's say you take a simple Fourier transform and you're able to, let's say you see a high pass spectral density in the delta band, 0, 1.0 to 3, then it means that the person is in a deep sleep or a meditation state. Okay, so theta would be during, again, it's also evoked during sleep. So during sleep, both delta and theta evoke. During relaxation phase, you will have your alpha rhythms which are being evoked, and beta is evoked when you do some mental task, and gamma is actually evoked when you do some high order brain activity. Okay, so from the recorded brain signals, we can roughly say what state the brain is actually in. Okay, so this is the sort of the main reason for recording brain signal. So if you have, let's say Mr. X or Miss Y has epilepsy, then the frequency ranges is going to be different, and then the amplitudes of the waveforms obtained in the time domain is also going to be different. Okay, so that is this is from a healthy person viewpoint. So these are the features or the parameters of the electroencephalogram for a healthy person. Okay, so but it's not always the case. So we know that there are a lot of brain disorders and neurological issues. So there could be epilepsy. The signal evoked for an epileptic patient will be different. So let's say a person who has stroke will be slightly different, right? So there are going to be a lot of variations in the electroencephalogram signals for the between the normal healthy person and between people who have psychiatric diseases. Okay, so this is sort of the let's say a tree diagram on the usage of the electroencephalogram. Okay. Okay, so again, I'm going to show this figure. So I'm going to touch upon your brain. So I just introduced you to the brain imaging techniques, and also I explained to you about, resting potential of a single neuron, action potential from, let's say, a bunch of neurons, and also explained to you the physiological basis of electroencephalogram. Okay, so now let's see what are the sensors which can be used to record this electroencephalogram, which is nothing but the signal which we want to monitor to study the brain. Okay, so usually the sensors or the electrodes are placed across the head. So let's say this is the head of a person, this is the ears, and this is the nose. Okay. So you first measure the distance between the nasal to enian, and then you distribute your electrodes, placing the first electrode at the 10%, and after then you traverse 20% of the distance to place the next electrode. You go all the way, and then you place electrodes across your head, and then you traverse this way in a similar manner, wherein electrodes are placed. The first electrode is placed after traversing 10% of the distance, and then from this side also 10%, and in between you place a electrode after you traverse 20% of the measured distances. Okay, so in this way you're placing your electrodes so that you are covering the entire brain. So you want to, let's say, record the signals from all the regions of the brain. So you can see the electrodes which are placed over here. So if it is an F, it means it's a frontal region. C means central region. P means parietal regions. And odd numbers indicate left side of your head or the brain, and the even numbers indicate the right side of your brain. Okay. So these are some commercially available sensors. So for EEG, so you have your gel based. So gel is actually applied to the scalp of the head, so that the impedance is actually reduced, so that the electrical recording, the summation of the electric potentials which we just discussed, so that the electrode picks up those potentials, right? So if you want the electrodes to pick up the potentials of the hair neurons occur at this point of the head, you want the impedance to be low, right? So for that, there are gel based electrodes. Active, in the sense, active has some circuitry onto the electrode itself, so that the noise is actually reduced when compared to the passive. I'll show you the difference between active electrode and a passive electrode. So this is the gel based electrode systems. This is a different sponge based passive electrode, and currently there is dry electrodes which are being sort of available commercially. Though the gold standard is the wet electrodes, dry electrodes are preferred when you want the signal over extended periods of time. Okay, so let's say you want to record a person who is having epilepsy for, let's say, 2 days continuously, right? And you want to allow him to go about doing his daily activity, then the gel based electrodes might not be good, because they would dry up and the signal would not be recorded properly, and it would have a low signal to noise ratio after, let's say, 1 or 2, after 2 hours. But the dielectrode can be used over extended periods of time. So again, the selection of electrodes or the sensors basically depends upon the application or the need you're looking for. Okay. So the difference between the passive electrodes and the active electrodes is shown over here. So let's say this is the head of the person. You're keeping an electrode, the passive electrode, and the active electrode on the head. And then there's going to be a cable before it goes to the amplifier, right? So the passive electrode, actually, there could be additional noises which sort of could be from the head movements or from the movements of the cable before it actually gets to the amplifier. So, but if you have an active electrode, so then this active electrode actually records the signal and sort of does intelligent signal processing because of the electronic circuitry which is there on the electrode, and there's not going to the and this connection is actually not going to affect much, because the signal is going to be directly transmitted to your amplifier. So potential interferences from the use of active electrodes is going to be minimum when compared to your passive electrodes. Okay, so these are all some electrodes which are available in the market. So mostly the passive electrodes are made up of AgCl. You can see the electrodes over here. So conductive gel or saline is usually applied between the electrode and the scalp of the human brain to reduce impedance. And subject hair is usually dry, and electrode site is usually cleaned, so that the signal recording is actually obtained in a neat and good fashion. Okay. So those are the wet electrodes and the active electrodes and passive electrodes. So dry electrodes, these are the evolution of the dry electrodes since 2010 up to 2015. I mean, even now, a lot of research is being happening as to how to get electrodes for electroencephalogram without the use of gel. Okay, so but still the level of signal quality which is obtained using dry sensors is not as good as your wet electrodes. So there's still a lot of work which still needs to be done. And this is sort of the evolution of the dry electrodes over time. So this is some of the work from 2023, which I managed to pull out so that it will be interesting for all of you. I think all of you are interested more in sensors. So there is some exciting work wherein graphene is being used to record the brain signals. Okay. So, I mean, unfortunately, I am not into that area of research in sensors, but I'll just give you a brief idea so that you can go back and read and see if it's relevant. And if it's something relevant and you think it would be exciting for us, you can always email me and also get back to us so that we can have more discussions in future. So it will be more like you teaching me than me learning, me teaching you in terms of the current exact graphene-based sensors and also nano sensors. Okay, so over here, the same sensors which I showed you, I mean, graphene is being used to record your signals over here. So these are some the placements of the electrographene electrodes onto the scalp of a person. But I mean, but the paper actually shows these figures. So it's early days for graphene-based sensors. So you see, this is your wet 8-channel normal wet sensor electrodes. This is a graphene base. It's the noise signal to noise ratio is poor when compared to the wet sensor based system. So you can see the SNR depicted over here. This is for the AGCL sensor, and this is for the graphene sensor. So the signal to noise ratio obtained over here seems to be much higher. So I think there's still a lot of work for all of us to pursue to make to overcome this gel-based sensor recording for electroencephalogram. So one other an interesting paper which I saw from 2021 was a sensitive and specific nano sensor for monitoring extracellular potassium levels in the brain. So over here, as we discussed in the earlier slides, so if you want to monitor the potassium levels at a neuronal level, so they probably have used a specific nano sensor over here. Okay, so you can probably go look up this publication for more details. I mean, I'm not exactly sure about the technicalities of it. I just breezed through the publication, looking at the figures and abstract and the conclusions, and I found it interesting. But again, it was at a mouse with a mouse experiment wherein they were able to monitor the potassium levels in the brain using a nano sensor, which was, I think, sort of doped or sort of modulated with some K plus fluorescence technique. Okay, this requires more reading for myself as well, so I'm not going to give too much about this. So I'll just give this as a food for thought for all of you to go back and read and understand. Okay, so I've actually coming back again to the same bird's eye view slide which I told you. So now we finished all these parts. So now we are going to look at how to actually realize a brain-computer interface system. Like, let's say you want to spell a letter without typing. So you want to spell a letter just using your brain signal. So how are we going to do that? So that's going to be the next 5-10 minutes. Okay. So what's a brain-computer interface? It actually enables, it was actually started off as a clinical application to give a way to enable severely paralyzed people to communicate without depending on muscle control. Okay, so but with time, so that actually that technology was used to sort of extend to other devices, and let's say to it was, let's say, to move a wheelchair or let's say to spell, right? So a lot of host of applications have actually popped in using BCI of late. So what does BCI actually combine? So BCI needs hardware and a cognitive paradigm. So cognitive paradigm is very important. So you need to evoke the brain states, right? So you need to evoke the brain states, and cognitive paradigm is the stuff which actually evokes the signals of your brain so that you can decipher what exactly is happening within that scenario for your machine learning algorithms to pick and translate the signals to command. Okay, so I hope this will be clear as I get into the slides and discuss more about BCI. Okay. So over here, you can see the entire system. So this is what is the electroencephalogram, Mr. X or Miss Y. He or she is doing the experiment. So there is a EEG cap, which is sort of over here, and there are electrodes which are on the scalp of the head. And as I said, each electrode is going to measure the synaptic potentials of a bunch or a ensemble of neurons, right? So once that is actually measured, so that's going to be some pre-processing, feature extraction, and classification. So what I'm now going to explain to you is this cognitive paradigm. How do we actually set up an experiment so that we are able to spell the letter H I, right? GH. So we want to spell this letter using brain signals. So that actually is what I'm going to show over here. So let's say we are collecting a single electrode. Okay, a single electrode, and we are let's say we are displaying the letter the same. We are going to display this matrix, right? In this matrix, we are going to flash this as one row, and then after some time we are going to flash this column, and then we are going to flash this row. So there's going to be alternate flashing of the row and the columns for the subject to view, and the subject has to count the number of columns or the rows where the letter H actually flashes. So when this row is flashed, because he wants to spell H, he needs to count it as one. Okay. And after the first flash of this row, let's say this row is flashing, he would not count because there's no H in this row. So then again, let's say if this column is actually flashed, the letter H is actually there in this column. So he would count as 2. So he keeps a track of the rows and columns corresponding to the letter he wants to spell as the flashing happens. So when that flashing happens, because H is actually the word he wants to spell, right? So because he's focusing on it, so there's going to be a potential which is evoked after 300 milliseconds. Okay, so let me give you an analogous scenario for this. So let's say you're waiting at, let's say, an airport or railway station for your mother or let's say your son, right? So your entire focus is going to be whether your mother or son is going to come, right? So you're not bothered about other flashes or other people. So you're all waiting for your mother or your son. So whenever you see your mother or your son, you're going to excited, and your brain knows that, yeah, my mom has come, my son has come, right? So that is the basic principle of the P300. There's going to be a potential which is evoked 300 milliseconds based upon the task which you're doing. So the task over here is you're focusing on the letter H, and whenever there's a flash of the row or the column correspondingly where H is there, there's going to be evoking. So in the airport or the railway scenario, whenever you see your mom or your son, there's going to be a evoking of potential after 300 milliseconds. Okay. So that is what is the P300 paradigm. It's an event related potential, right? It's to a definable reference event. So the reference event over here is the occurrence of the letter H. Okay. So it's a temporal response to the stimuli. I'll show you the experiment. It's a visual stimuli which we have shown to a subject wherein he spells a letter. And so this is sort of going to be as he spells for one particular so this is the display time of that flash, and this is the time between two flashes, and you keep on recording your signals as these flashes go on. Okay, so you can see I'll roughly show you the experiment. So so the target letter is R. R is first displayed. So you can see the R is displayed over here, and there's going to be flashes of rows and columns. Okay. So whenever the row or the column where R is present, the subject counts that in his mind. Okay. And because of he's focusing on the letter R, there's going to be a P300 potential, which is evoked 300 milliseconds after the flashing of the row or the column corresponding to R. So after some time, you can see that the system will show R from the set of the signals which are recorded. Yeah, so you can see the system has predicted the letter R. Okay. So there are various other cognitive paradigms. I don't think I'll have time for it. So I will just quickly show another one, the SSVP system, which was developed at my alma mater. This was done with my supervisor and another good friend of mine. So over here, you can see that he's trying to control the cursor using his brain signals. So the four squares are actually at a different frequency. So if he wants to move the cursor up, he will focus on this part of the cursor. And if he wants to move the cursor left, he will focus on this. So then the cursor will move to the, right. So if it's going to move to the right. If he wants to move it to the bottom, he's going to focus on this part of the square, which is at a different frequencies. So then this will move down when he focuses at the bottom part of the square. So the three squares are actually having stimulus at different frequencies. And when the person focuses on one side of the square, a corresponding frequency is evoked into your brain, which is detected by the system, and it is passed as a control word or a control command so that the cursor moves automatically. Okay. So I think hopefully I'm clear in explaining this to you. So these are some other applications. So what started off with medical application to communicate for a person who was completely logged in. Now there's a lot of applications for those, let's say neuromarketing. You want to see, let's say, which portion of the advertisement is actually evoking more excitement in the audience, right? And it's, let's say, even in education, let's say if someone's listening to lectures, is the person listening to the lecture or not listening to the lecture? So applications like those have popped up in the recent past. Okay. So I mean, I'll just quickly run through some work which we are doing at IIT Guwahati. So we are actually looking at EEG experiments for natural environment for healthy people. So let's say these are let's say someone's. Yeah, so just last moment. So if someone wants to get their brain signals recorded when they are on the move during the daily life activity, right? So we have a system wherein both the camera and the EEG data are being recorded, and we are trying to do some analysis on the smartphone. Okay. So that brings me to the end of the talk. I would like to thank all my lab members for for the creative ideas and for the hard work. So this is the neural engineering lab at IIT Guwahati. If anyone's interested in this type of work, please feel free to reach out to me. You can contact me at this email. And at the moment, we are looking for interns on circuit design and electronic components. So if anyone's interested in that, please do reach out. There's a special issue going on biomarker identification for neurological diseases. So if anyone's interested, please do submit. I would like to acknowledge the Spark grant, DST grants for funding our research. And I would like to once again thank Center for Nanotechnology for giving me this wonderful opportunity. If you have some questions, I'm glad to take them. Thanks a lot. I hope you enjoyed the talk.
Thank you, sir, for such an interesting talk. We'll take questions from the participants. So the first question is, can you please explain again what action potential means?
Sorry, can you come again?
Sir, what does the action potential mean?
Okay, so let me get back there. So can you see my slides?
Yes, sir.
Okay. Okay, so let me go slowly. Action potential is actually generated at the axon trigger zone. Okay, so let's say you have a lot of signals coming in through the synapse. Okay, so that is going to be action potential is going to be generated when it crosses the threshold potential, which is shown over here. Okay, so that could be due to the depolarization or the, let's say, entry of Ca2 plus or Na plus. Okay, so when that all this happens at the same point, right? So let's say this is one NaCa2 plus, this is Na2 plus, and let's say another synapse with another high depolarization happens. So this is all going to get summed up, and this will actually create a action potential which actually passes through the axon of your neuron, of a single neuron. That's what I showed over here. Oh, sorry, one second. Huh. So that's what was shown over here. So action potentials are generated when all the depolarizations cross the threshold. So you don't see a action potential for the hyperpolarization current. You see a action potential only after depolarizations, right? You see the depolarizing current which causes the depolarization of your neuronal membrane. And when these sum up and it crosses the threshold, there is an action potential which is generated. I hope I was able to answer that, and it's clear. Please let me know if it's still not clear.
Yes, sir. Thank you, sir. Due to lack of time, you are requested to address the rest of the queries in the chat box.
Sure,
Thank
sure.
you so much. Thank you for the
Okay,
talk.
thank you. Thanks a lot.
Moving on to our next talk, we have Dr. T. Malleshwar Rao from IIT Guwahati. Dr. Malleshwar Rao acquired his PhD from Jawaharlal Nehru Center for Advanced Scientific Research, JNCSR, India, and is currently a professor at Department of Physics, IIT Guwahati. His research interests broadly lie on quantum materials, semiconductors, epitaxial thin film, nanowire, spintronics, optoelectronic switching, spin orbital, non-collinear spin textures. Currently, his research emphasizes on exploring the fascinating realm of quantum materials and low-dimensional physics, such as phase-coherent quantum transport, quantum confinement effect for exciting applications, such as energy-saving spintronics, optoelectronics, and quantum computing. Today, he will be delivering a talk on Group 3 nitrides for optoelectronics, QCSC in nanowires LED. We now like to request sir to kindly take over the session. Over to you, sir. Sir, you are not audible. Sir,
How about now?
yeah. Now it's audible.
Okay. Thanks for your kind introduction. My name is Malleswara Tangi. I am an assistant professor in Department of Physics, IIT Guwahati. And I would like to thank organizing committee and especially Professor Arun Tej and Professor Pamu for giving me this opportunity to talk in online familiarization workshop at Center for Nanotechnology. And I would like to present today briefly about group 3 nitrates, how they are utilized in optoelectronics, especially for light emitting devices. So outline of my talk as follows. First, briefly, I discuss about semiconducting technology and various applications and how these semiconducting materials can be prepared by using various growth methods. And my focus will be on molecular beam epitaxy grown semiconducting materials and LED devices and how they will be characterized, and eventually I will talk about nanowire based light emitting devices and optoelectronic devices and also I will discuss about visible and UV LEDs and efficiency droop. And also I discuss about piezotronics and finally the integration of 3D and 2D bulk and two-dimensional semiconductors for realizing quantum well structures. So as we know, we have like advanced from the Stone Age to electronic age. Electronic age is mainly because of advancement in the semiconductor technology. As you can see here, especially from group 3 nitride semiconducting materials, they can be utilized light emitters such as LEDs and lasers. Also, they can be used for electronic devices like high electron mobility transistors and nano generators for clean energy applications in self-powered devices and clean energy water splitting applications. Also, these light emitters can be used in optical wireless communications. So in order to achieve these technological applications, one should have like a proper materials. Either it can be semiconducting, magnetic, or battery materials. In order to achieve that, we should have a crystal growth. Crystal growths are few types like melt, solution, vapors, and solid methods. By using melt method, we can grow bulk crystals using Bridgman and Czochralski method, where we can use this bulk crystal method, where we use a seed layer and by putting melt on that from supercooling this melt, we can get bulk crystals, which from bulk crystals we can thin them down and use them as substrates. And other important method is vapors. Using this method, we can grow thin films and nano to micro dimensional miniaturized materials, which are like we can call it as epitaxial semiconducting materials. Using this method, there are two methods. One is chemical vapor method and physical vapor method. Under chemical vapor method, using chemical routes like chemical vapor deposition and metal organic chemical vapor deposition, we can grow these thin films. And other one is physical vapor method, where we use by means of heat, electron bombardment, laser ablation, or vaporization of metals. All these methods come under physical vapor method. For example, sputtering, e-beam evaporation, pulsar laser deposition, and molecular beam epitaxy. My focus will be on molecular beam epitaxy, and it's like how we use molecular beam epitaxy to prepare these technologically important materials. So molecular beam epitaxy, as I discussed, it is a growth technique. It is invented in 1968 from Bell Labs. And epitaxy is nothing but when we grow material on substrates, it has to grow in a well-ordered manner, and it has to mimic substrate crystal symmetry and sometimes crystal structure. And advantage of molecular beam epitaxy is it is UHV system, and the mean free path of atoms in this system would be way higher. And thus, when we evaporate any material, these atoms will not collide each other before reaching to the substrate. So that is the main advantage. Because of this advantage, we can have fine control over growth parameters. Like we can vary the source temperature in a fine manner, and you can deposit atom by atom. And also you will get best uniformity and best material quality. Apart from this, we can use characterization tools to online monitor the growth, such as read BFM, QCM, RJ, and ellipsometer. Also, cathodoluminescence, such in situ characterization tools we can use in this system. And this is well suited for scientific research. Nowadays, there are industrial scale molecular beam epitaxy systems as well. So uh, you can customize these molecular beam epitaxy system from research scale to industrial scales. So using this molecular beam epitaxy, we have several growth modes. Like when we evaporate these materials, they impinge onto the substrate. When it will have several processes like absorption, desorption, interdiffusion, and clustering of these atoms. And a nucleation to be happen, it has to like this surface free energy, interface free energy, and surface free substrate free energy, film free energy, they should obey this relation. Mainly substrate free energy should be less than interfacial and film free energies, then only nucleation happens. And this nucleation are three different types. One is Frank van der Marwe, where layer by layer growth takes place. When you have film lattice parameter is exactly substrate lattice parameter like homo epitaxy kind of growth, you will have a layer by layer growth. And next one is Walmer Weber growth. This is island growth. When you have lattice mismatch, very high lattice mismatch between substrate and film, this growth takes place. And the other one is Stransky Krastenov growth mode. This is layer by island. Initially, it forms a layer by layer growth mode, and then it switches to a layer by island growth mode. By using this growth mode, we can grow quantum dots. And by using Walmer Weber, we can grow nanowires or island kind of growth. So as I discussed, read can be used to in situ monitor the growth. Here by there will be an electron gun where the electron beam incident at grazing angle onto the film, and the diffraction pattern will be collected on the fluorescent screen. Based on this diffraction pattern, we can decide whether the growth is layer by layer or nanowire or island kind of growth. For layer by layer smooth growth, you will have perfectly streaky pattern. For island growth, these electrons scatter at the islands, and you will have a spotty kind of read pattern. So why do we focus on group 3 nitrates for optoelectronics? Because here, as you can see in this figure, band gap is plotted versus lattice constant. Uh, Here, interestingly, as compared to other semiconducting materials like second and sixth group and third, fifth materials, these group 3 nitrates, these are also third, fifth materials, but only nitrates. These span a very large wavelength regime like deep UV 6.2 eV aluminum nitrate to indium nitrate 0.7 electron volts. It has huge span of wavelength regime as compared to other semiconductors. This is main advantage. Within the same nitrate system, you can have all the wavelength emissions from these materials. And gallium nitride is very important material out of these materials, and it has band gap of 3.4 electron volts. In terms of wavelength, it will be around 364 nanometers. Among these nitrates, initially indium nitride had some band gap issue because researchers understood it wrongly because of poor growth techniques such as sputtering. Initially, people believed that indium nitride has very high band gap because of synthesis problems like high vapor pressure of nitrogen. It has low dissociation temperature and lack of native substrates. And this high band gap is attributed to initially oxygen inclusions, quantum size effects, and Moss-Bernstein effect. Here, Moss-Bernstein effect is nothing but when you have a heavily doped semiconductor, Fermi level shifts into the conduction band, and hence, if you do absorption or photoluminescence measurements in such case, you will have a higher absorption edge. Like absorption edge will happen at higher energy. So this is called Moss-Bernstein effect. Because of the band filling effects, Fermi level shifts into the conduction band, and hence you will see higher band gap. You can see the literature values how they have how they scattered, and this is all because of the synthesis problems. But after achieving advancement in the growth techniques like MBE and MOCVD, after mid 1990s, people have achieved very good electronic properties like high electron mobility and low carrier concentration for this semiconducting material. This also plays crucial role when we prepare indium gallium nitride, which is active region material for optoelectronic LEDs or lasers. Then further, one more problem is dislocations or other defects in these films. By employing nano E-log, like epitaxial lateral overlayer growth, by leaving voids at the interface, we can reduce dislocations in the bulk region of the films. So that way we can reduce dislocations, and hence you can get better optical or electronic properties. So as compared to other semiconducting materials, gallium nitride exhibits very high density of defects like dislocations, as you can see in this figure. Just give me a moment. As you can see in this figure, you have several defects as compared to zinc selenide, whereas zinc selenide TEM cross section doesn't show many defects, but gallium nitride has a lot of defects, and the range is something up to 10 power 9 per centimeter square, whereas for zinc selenide it is 10 power 2 to 10 power 3 per centimeter square. Even then, irrespective of these dislocations or other defects, gallium nitride relatively shows better properties in terms of like optical emission as compared to other second, sixth, or other third, fifth semiconducting material. This is main advantage of group 3 nitride system. So as you can see in this figure, efficiency versus dislocation density. Here, at even the other materials have low dislocation density. They have efficiency uh, relative to the gallium nitride, but gallium nitride shows a very high dislocation density. Even then, it has similar efficiency ranges. This happens for gallium nitride and aluminum gallium nitride, but if you take indium gallium nitride case, even it exhibits even more than 10 power 9 dislocation density. It exhibits better efficiency than other materials. This is also one of the advantages of nitride system. So as compared to gallium nitride and aluminum gallium nitride, indium gallium nitride shows better efficiency if we prepare LEDs out of this material. So what is LED? When you prepare PN junction out of same semiconducting material system, at the junction, if you pass current through the PN junction, at the junction you will have a carrier recombination, electron and hole pair recombine and emit light. This is main principle behind LED operation. So you have to have a P-type and N-type material. First you need to have intrinsic semiconductor, and you have to dope the material in order to get p-type and n-type materials. Sometimes for some materials doping is a big issue. Gallium initially like Suji Nakamura faced a big problem doping gallium nitride, especially a p-type, achieving p-type gallium nitride. He used a two-flow method of CVD, where he used magnesium as dopant. Initially, those P-type dopant materials are highly resistive. Then he realized that if we post anneal the samples, this P-type material conducts well. And then he utilized this P-type magnesium doped gallium nitride as P part of the PN junction. So here, as you can see here, he used p-type and n-type junction, and in between that InGaN quantum well has been used. InGaN quantum well as active region, by changing indium composition of this active region, you can tune the emission wavelength from gallium nitride band gap 3.4 to indium nitride band gap 0.67 electron volts. But the problem is, when you incorporate more indium, it will have some other issues. We will discuss that as well. So this is brief history of light emitting diodes. In late 1980s, first people have observed electroluminescence on silicon carbide material. That is the main origin. Then people have explored several materials like phosphide, arsenides, and selenides. especially for with gallium arsenide and gallium phosphide materials. In 1962, Professor Holonaik tried to grow this gallium arsenide phosphide materials, and he could achieve a very dim red LED emission from his device. However, he called as father of LED. He could not get Nobel Prize because of his low efficient devices. Then Professor George Crawford, he tried to use similar system, gallium arsenide phosphide, and he could achieve both green and pale yellow light emission. So it took 10 years to get green and yellow and then red LED emissions. But afterwards, in order to get white light, we have to mix green, red, and blue. But getting blue at the time is very difficult because there is no good material system. However, people find gallium nitride is good material. It is difficult to grow. There are some hurdles to prepare high quality gallium nitride material. Then after nearly 20 years, Shuzi Nakamura realized that gallium nitride can be grown in a two-flow CVD method, and he could achieve very high quality crystals. As I discussed, though he achieved high quality gallium nitride crystals, he was having problem in doping them. Though he got N-type material, it was difficult to grow magnesium doped P-type gallium nitride. Then he, by post-annealing methods and hydrogen passivation methods, he eventually achieved P-type gallium nitride as well. And first InGaN blue LED he made out of this 3 nitride system, and it is highly efficient as compared to all the LEDs like found by the time. So once he achieved blue light LED, so we have to get white light from all these LEDs. But the problem is the green and red LEDs are from other material system, and he has to still work on that. And in order to get white light, he has to tune indium composition so that he will get green and red emissions as well from the same material system. So he tried to tune indium composition in order to get green and red emissions. But what he observed is by increasing indium incorporation in InGaN material, he observed emission intensities quite low as compared to low indium content. So that also gives another problem when you want to have white light emission. So these all these material systems have a green gap. Like when you have nitride system, when you move from blue light to green emission, this efficiency decreases. If you take a phosphide system like gallium arsenide phosphide system, when you have more phosphorus into it, it will emit around the green gap. But this material becomes indirect band gap when you have more phosphorus into it. So because of these two problems, in the green wavelength regime, green LEDs have very low efficiency. So this is the problem when we want to have mixing these blue, green, and red LEDs. Since green LEDs are low efficient, it is difficult to get high efficient white light emission out of this mixer. So that is one of the problems. And the other problem is this is intrinsic material problem. It is called efficiency droop. When you have all these LEDs, if you have to inject current to get high emission, like high luminescence of light. So while increasing current, what happens is this external or internal quantum efficiency decreases. So as you can see in this figure, increasing current gives like a decreasing in efficiency. For UV LED, it is 11%, blue LED 40%, and for green LED, it is 54%. And UV LED has relatively high turn on voltage as compared to blue and green LED, because UV LED uses aluminum gallium nitride, which is a high band gap material, and its resistivity is relatively higher than blue and green material. And finding ohmic contact for such material also is another issue. So another in this figure, as you can see, when you inject carriers into the device, they will recombine at the active region, which is quantum well or multi-quantum well region. And these recombinations are three types. One is radiative recombination, other one is Shockley Reed Hall or Ose recombination, which are a part of non-radiative recombinations. And in addition to these recombination, some electrons can leak through the device. Like they may not participate in the recombination process. Simply they escape and go towards p-type gallium nitride. This is called as carrier leakage from the device. So because of this carrier leakage, we see this efficiency droop at higher injection currents. In order to minimize or overcome this issue, researchers have tried electron blocking layer just after multi-quantum well. But even after using this electron blocking layer is nothing but high band gap material, which is higher than p-type, n-type, and multi-quantum well or barrier layers. Even then, this stopping completely this carrier leakage problem becomes elusive. It is not easy to completely stop this carrier leakage problem. That's why nitride LEDs suffer from this efficiency droop a big time. And this any efficiency, it can be expressed as ABC model. As you can see, LED rate equation, when you inject carriers into the device, how like what ways they will be consumed in the device. And this is represented by ABC model. And A is Shockley Reed Hall coefficient, B is radiative coefficient, and C is Ose coefficient. And radiative mechanism, we know when the carriers recombine from edge to edge, like band to band, then it will give they will recombine radiatively, and it will give photons, only photons. That is good for optoelectronic devices. But there are other mechanism, recombination mechanism, which is non-radiative. In non-radiative recombination, other mechanisms like defects or intraband transitions or shallow or deep level defects will contribute to this non-radiative recombination process. And importantly, these non-radiative recombination mechanisms are Ose and Shockley Reed Hall recombinations. In Ose recombination, when like primarily recombination process between electron and hole happens, and that emission energy will be utilized for intraband transitions. Like within conduction band, electrons will like transit from one level to the other level, and they relax by emitting a phonon. So phonon means it will heat up the device. That's how it reduces efficiency. And the other one is carrier delocalization via a defect recombination process. When you have defect states like dislocations or point defects, interstitials in the semiconducting crystal lattice, they will they will include intergap states in between conduction band and valence band edge. If they are shallow levels, they may radiatively emit, but if they are deeper levels, they will recombine non-radiatively, and it is not useful for good optoelectronic devices. So we have to have only radiative recombinations when we use any material for LED or laser applications. And the internal quantum efficiency is nothing but radiative, radiative recombinations over total recombinations. And it can be expressed in terms of carrier density as well from ABC model, like radiatively recombinations over all recombinations. So this is efficiency evolution for LEDs. As you can see here, till late 1980s, there are materials like arsenides and phosphides which contribute to the LED research, and they have they achieved a high efficiency, but they took long time. But after 1990s, because of the invention of gallium nitride by Shuji Nakamura, it took in no time they have achieved as high as other materials, efficiencies as high as other materials like InGaN blue LEDs. Within 10 years they achieved high efficient LEDs. And green LEDs are relatively low efficiency, as I discussed. When you incorporate indium into the device, it will affect emission because of indium fluctuations in the material. And in the UV region also, aluminum gallium nitride had a very important role, and it has been utilized to get even deep UV emission as well by changing aluminum composition in gallium nitride lattice. So this is how we evolved in terms of using LEDs. Initially, we used to use oil lamps, which have very less luminescence per watt, as compared to LED. You can see like Thomas Alva Edison bulb or fluorescent lamps. They have a way lower luminescence per watt, and LED is much cheaper and energy efficient as well. And this is global lighting sales. So by 2020, we can see like 50% of like light sources are replaced by LEDs, as you can see in this figure. Maybe by now it must be crossed even 60 or 70%. So LED, we all know that LED have several applications like solid state lighting for decoration. Also, car headlamps nowadays use very high luminescence LEDs and LED displays, and it can be used for agriculture as well. If you have LEDs, you don't even have to have a sunlight for agriculture and for indoor lighting applications. So this is all a brief history of LEDs. I also contributed somewhat to this research. So now I am going to talk about nanowire LEDs, where I worked in this area I worked, and I will focus on quantum confined Stark effect in nanowire LEDs. Many people have reported that quantum confined Stark effect is observed only in thin films, but not in nanowires. But later we observed that even in nanowires, because of quantum barrier or quantum well lattice mismatch, there could be tensile or compressive strain within the active region and in the quantum well region. So this compressive or tensile strain will change energy levels within the quantum well region. So basically, when you apply electric field, the energy splits. That is called Stark effect. If this happens in quantum well region because of the strain or whatsoever, it is called quantum confined Stark effect. So we observe this quantum confined Stark effect even in nanowires. So this third nitride material system, they have a specific and unique property. They exhibit polar nature. These are available in woodsite or zinc blende crystal structure, but most thermodynamically favorable structure is woodsite material. Several researchers have tried to grow zinc blende structure, but they are not successful because it is difficult to get this phase, zinc blende phase. Zinc blende phase is good for optoelectronic devices because it doesn't exhibit any polar nature, and this polarization kills device efficiency. And even in woodsite materials, we can grow either polar, semi-polar, or non-polar substrates, and we can get non-polar material. But relatively, growing polar material is way easier than semi-polar or non-polar materials. And advantages of this polarity is we can study piezotronics, and also because of this polar nature between gallium nitride and aluminum gallium nitride interfaces, they will have a two-dimensional electron gas which were utilized as high electron mobility transistors. And also this polar nature gives p-type carriers when you have UV wavelength algan materials. As discussed, main disadvantage is that it reduces rate of recombination of carriers, and hence like it gives low efficient LED devices. I utilize molecular beam epitaxy. It is state-of-the-art growth facility. It is UHV system and pumped by several like pumping systems like cryo pump, ion pump, and turbomolecular pump to achieve 10 power minus 10 even low pressures. We utilize effusion cells in order to evaporate group 3 materials, gallium, aluminum, and indium. For nitrogen, we use nitrogen plasma source. We grew these nanowires on silicon substrates on silicon 111 orientation. Before loading in silicon into the chamber, we clean that in a special way using dilute HF, and then we introduce that into load lock chamber and outgas at 200 degrees centigrade for 30 minutes, and then buffer chamber, and finally in growth chamber we heat up the substrate till 900 degrees centigrade, and then slowly cool down. While cooling down the substrate, we observe very famous 7 by 7 reconstruction. This confirms that removal of native oxide from the substrate, because any substrate it can have native oxide. So to get pristine surface of the substrate, we have to remove native oxide. So this can be done in MBE by heating up the substrate up to 900 degrees centigrade. And we grew gallium nitride first, n-type gallium nitride, by doping silicon into the material at 700 degrees centigrade. And gallium and indium flux were calibrated using beam flux monitor by noting down beam equivalent pressure values. And the nitrogen plasma conditions are nitrogen flow rate is 1.5 SCM at 350 watts. When we use nitrogen plasma source, the chamber pressure will be a bit higher, around 10 power minus 5 torr. And the dopants are magnesium. Magnesium is evaporated using low temperature effusion cell, whereas silicon is evaporated using high temperature effusion cell. And the indium composition in the active region is around 37%. So this is for silicon 7 by 7 reconstruction, and this will be like streaky pattern as I discussed earlier. It comes on perfect layer by layer growth, and on nanowires we see this kind of spotty pattern. Actually, there are some other methods using vapor, liquid, solid phase epitaxy or selective growth epitaxy. but it consumes a lot of effort. You have to make a template having some holes into the substrate. They will act as they will help to nucleate material in the form of islands initially, and then they help to grow nanowires. But using our process, like in MBE, using nitrogen rich conditions, without having all these time consuming processes, we can directly grow very high quality nanowires, having very high quality and high density and uniformity. This is FESM top view, and this is FESM cross sectional view of nanowires on silicon. And then we have to determine the polarity. Since these are polar materials, it can be either gallium polar or nitrogen polar. For gallium polar materials, bond direction will be gallium to nitrogen along C orientation of the substrate, or 111 orientation of silicon. For nitrogen polar, it will be like bond direction will be nitrogen to gallium, which is represented by 0001 for nitrogen polar case. Experimentally, we can determine the polarity by two ways. One is valence band spectrum obtained by high resolution X-ray photoelectron spectroscopy. In the valence band spectrum, you will have a shallow states which come from shallow levels like 1s or p states, and based on the intensity ratio, you can determine the polarity of grown nanowires. And the other way is by etching chemical routes. If we etch these nanowires by using KOH, if they have needle-like shapes, it will be nitrogen polar. If they have flat surfaces like pristine nanowires, it will be a gallium polar. Since we obtain needle-like shapes, we confirm that it is nitrogen polar crystal. And also from valence band studies, this also corroborates that it is nitrogen polar material. Then to understand its structural property, we did HADAF STM and high resolution X-ray diffraction measurements. As you can see in this image from STEM, you can clearly see 10 quantum wells within the nanowire, which are sandwiched between N-type gallium nitride and P-type gallium nitride. And the barrier layer is gallium nitride and disk. So this is quantum well when you talk about thin films. Since these are embedded in nanowires, we call it as quantum disk instead of quantum well. So this quantum disk is from indium gallium nitride having thickness 3 nanometer, and barrier layer has thickness of 7 nanometer. And actually for like any material to exhibit quantum size effects, it should be less than its Bohr exciton radius. For InGaN, it is around 4, for gallium nitride system, it is around 4 nanometers. So if we have a quantum well layer within like less than Bohr exciton radius, it will have quantum size effects, like quantum confinement effects. Then we did selective area electron diffraction, which confirmed that these crystals are Woodside crystalline in nature and highly single crystalline oriented crystals. From yields, electron energy loss spectroscopy, elemental mapping, we identify that the indium is incorporated within the quantum well region. And further, we also did HRXRD to confirm its crystalline quality and crystal structure, and it belongs to hexagonal Woodside crystal structure. So we could grow very high quality nanowires having highly single crystalline oriented and high crystal quality material. Now we want to study these materials, how good they are optical in terms of optical characterization. For this, we did temperature dependent microphotoluminescence, with excitation wavelength 405 nanometers, which is blue excitation with blue LED, a blue laser diode, sorry. So here we did temperature dependent studies. As you can see here, the intensity ratio, integral intensity ratio at room temperature and 10 Kelvin, it gives around 58%. This is also like a relatively better quality than state of the art LED materials. And further, we studied that using Versionist condition, we confirmed that there are no tail-like band states at the band edges of quantum well region. Once we confirm that these nanowires are structurally and optically very good materials, we have gone for clean room device fabrication. As I discussed, we first grew high quality nanowires using MBE on silicon 111 substrates. And then we deposit perylene. This is a polymer which has a very high transmission in visible and UV region, and also it helps to it is kind of insulating material. It will shorting top and bottom electrodes. So for that purpose, we deposited perylene. We planarize these nanowires. Then we etch away this perylene till we see top p-type gallium nitride nanowires. We used reactive ion etching process for this. Then we deposit nickel ITO. By rapid thermal annealing, we can achieve a transparent ITO on top of these p-type nanowires. And it also acts as a current spreading layer. Like when you inject current into the device, it will uniformly inject across the device if we have a highly conductive nickel ITO deposition. That is the role of nickel ITO deposition here. Then we did photolithography, mesa etching, in order to get devices areas having 500 micron by 500 micron, and eventually we deposit metal pads on top of these mesa etching devices. So this is eventual device structure where you have a silicon substrate. Backside you have a metal contact, aluminum and gold N-type contact. And on p-type material, we have nickel ITO and tie gold contacts. And thicknesses are as shown in this image. Then we did electrical characterization. We tested these devices. In LIV characteristics, you can see that as increasing a forward current, injection current, output power increases, but it has as you like, if you see this voltage versus current, it has slightly higher turn on voltage around 2.5 volts. But for state of the art LED devices, this is way lower. The problem this problem arises because of unavoidable silicon nitride formation during MBE growth process. Since we use nitrogen plasma source in molecular beam epitaxy system, the silicon has a high tendency to form silicon nitride as interface layer just before growing gallium nitride. And the silicon nitride layer acts as barrier, kind of barrier, so electrons tunnel through. So that results in this high turn on voltage. Except that all other properties are as good as like thin film LEDs. Here we see a low output power because we haven't used a integrated sphere in our characterization testing system. That's why we see relatively low output power. Then we inject current and try to see luminescence properties of these devices. With increasing current, we have slightly bluish blue shift in the emission that we will discuss in upcoming slides. And also with increasing current, as seen here, output power increases. Similarly, with increasing current, EL peak intensity increases. Also, this peak has slightly blue shift here with increasing carrier injection. So all this effort is to study quantum confined Stark effect in these nanowires. So when you have a polar material, the a total polarization will be from a spontaneous and piezoelectric polarization. If you compare spontaneous and piezoelectric piezoelectric polarization will have dominant values. From elastic theory of a semiconducting material, if you substitute all these elastic coefficients, and for indium gallium-nitride quantum well and gallium-nitride barrier layers, you will have tensile strain at the interface. This will give you positive value of the strain, and all these constants will give you a positive. So eventually you have positive value for this polarization. Positive value means it has to be around a gallium polar direction, from gallium to nitrogen bonding. And our crystals are grown along nitrogen polar direction. So this polarization of this material is like opposite to the nitrogen polar direction. So electric field will be opposite to polarization direction. So hence we can understand the direction of electric field inside this material. So why do we observe quantum confined Stark effect in nanowire LEDs? Means this is main reason, like because of these strain induced fields, this electron and hole they do not overlap. They will have some slight offset, and this causes for non-radiative recombination centers within the active region in quantum wells. So when you have this non-radiative recombination and uh, like non-overlap of electron and hole wave functions, you have reduced efficiency in the devices, and also you will have redshift if you do EL electroluminescence, cathodoluminescence, or photoluminescence. Even in optical absorption measurements, you should have redshift when you have quantum confined Stark effect in the material. So to understand this quantum confined Stark effect, we have employed bias dependent microphotoluminescence using excitation wavelength 473 nanometers with blue laser. Here, as you can see clearly, this is 0 volt. That means without a bias. This is what we observe. In forward bias case, we clearly see blue shift, and in the reverse bias case, there is a red shift. As I discussed in terms of elastic properties and strain within the quantum well, when you have forward bias for nitrogen polar crystal, piezoelectric field will be opposite to the forward bias. Hence, they will be compensating each other. That's why we see blue shift in the PL emission when you have forward bias. In the reverse bias case, it enhances quantum confined Stark effect. So we see a red shift in the PL emission. So by this, we understood that in order to get high efficient devices, we have to grow material along nitrogen polar direction. Gallium polar direction will not help. And same thing is studied by power dependent microphotoluminescence. Here we increase a power of the excitation source. With increasing power, again we observe a blue shift in the PL emission, which means that these photo generated carriers, they screen piezoelectric fields. Hence, you observe blue shift. Basically, it suppresses quantum confined Stark effect. And we also understand that because of this linear relation between intensity and power, it confirms that there are no localized states in quantum well, which are called bound states. So, and thus we clearly understood that this quantum confined Stark effect dominates in forward bias. If you, it will be have predominant effect if we have gallium polar crystals. So next one is till now we discussed about quantum confined Stark effect in visible LEDs. We also put some efforts to grow ultraviolet LEDs by using aluminum gallium nitride as quantum wells with different compositions. We can tune a UV wavelength as well by using aluminum gallium nitride. Likewise, indium gallium nitride in visible region. So these are aluminum gallium nitride nanowires, which have multiple quantum wells, multiple quantum disks basically, since these are in nanowires, having aluminum gallium nitride with aluminum more composition as barrier and less composition acts as quantum disk. And we fabricate devices as discussed in previous slides. And here what we did is we used a focused ion beam to make a pattern around the device to study like efficiencies, like how efficiencies would be with and without DBR pattern. And we try to simulate a DBR reflectivity having a known thickness of air gap between nitride and air thicknesses. And we choose these thicknesses such a way that this reflectivity matches emission wavelength. And this is a typical device structure. This is AFM image around the DBR pattern. And for aluminum gallium nitride, we did ellipsometry to have good idea about refractive index. So as you can see here, this nitride 8 dBr one is blue one, and the red one is control, like without dBr. So with dBr, we could see a low FWHM of electroluminescence emission, and similarly it has higher optical power and also higher external quantum efficiency as compared to without dBr pattern around it. So basically dBr helps to have like better light extraction efficiency. On another work, we also utilized this material for piezotronic and piezo-phototronic effect. In this study, what we did is we grew titanium on sapphire and titanium over tantalum nitride, and on top using that template we grew indium gallium nitride, but these are heavily doped by silicon, having carrier concentration around 10 power 9 per centimeter cube. So such materials we used to study piezotronic effect by using conducting AFM microscopy, where we utilized a platinum iridium tip to exert strain on the nanowire and study IV characteristics by increasing strain. Okay, I'm almost done. So you have strain effects in this semiconducting material, and it is called piezotronic effect. It is studied under photo excitation. Hence we studied, it is called as piezo-phototronic effect. And we also try to integrate with TMDs, and we realized that using aluminum indium nitride as barrier layers, we can have MOS to quantum well, which has blue shift in the photoluminescence spectra. So overall, this is the like a story of LEDs and also my research activities so far I have done. And I would like to sincerely thank all the research opportunities I got from previous institutes and Humboldt Fellowship, and also IIT Guwahati to have my position here. Thanks very much.
Thank you, sir, for such an interesting talk. Now we'll take questions from the participants. So the first question is, can you explain how thickness monitoring is done by the crystal oscillator in suit with NVD?
Yes, actually, we can monitor monolayer by monolayer. When you have like read intensity, when the film grows layer by layer, that intensity changes. It will be oscillatory function. So if you fit that oscillatory function, you can determine like monolayer thickness, like growth rates. You have to get intensity oscillations from uh, read images, deflection high energy electron diffraction images.
Thank you, sir. Sir, he's also asking, do IITG have MBE for outside use?
That we have done, sir. Yeah,
Sorry. Can you repeat it again? Which
Do
one is that?
IITG have MBE for outside use?
Outside?
For the external use. They're asking if they can access that.
He's a not member. We have done, sir.
Okay, so like we do not have.
We have a organic molecules only we'll be able to do it, not other molecules here.
Yeah, molecular beam epitaxy has like other materials also can be evaporated, both organic and inorganic materials. That depends on how we choose the temperature of sources. If I understand question correctly.
Skip the question.
Okay, sir. Sir, the next question is, the cost of indium is high. We need to choose alternate materials. Can you comment on this, sir?
Cost of indium or indium
Yes.
is not that cost. Indium is highly abundant material.
Sir. Manually silicon is cleaned by HF or by other mechanism. Handling HF is suggestible manually. How we remove the fluoride ion residue in the chamber? It's regarding the silicon in the chamber.
From chamber, which residue?
The
Which
fluoride
ion
ion.
residue?
Fluoride ion.
Fluoride ion.
Yes, sir.
Okay. Actually, we rinse them in deionized water before introducing into the chamber. And also we clean that in IPA, acetone, and then rinse like few times in deionized water, and then we introduce that in low log chamber. There should not be any HF remaining by the time. And if there is some HF, it should not affect. Like fluoride atoms. There should not be because we highly rinse that in deionized water.
Thank you, sir. Thank you so much for the talk. Sir, rest of the question, please, you can address them in the chat box.
Yeah, sure.
Thank you so much, sir.
Yeah, thank you.
We will
Can I
have a
leave?
short. Yes, sir.
Sir, there are some more questions in the chat box. If you can
Yeah,
answer
I will
them.
answer them. I will answer
Yes.
them. Yeah.
Yes.
Okay. Thank you, sir. Thank you so much for the talk. We will have a short tea break for 15 minutes. Participants are requested to come back by 12:15. Thank you very much.
Sure.
The map are you all in? Hello, ma'am. Are you online? Sorry, ma'am. Hello, ma'am. Okay. We will resume in 5 minutes due to some technical error.
Thanks, Andre.
Hello, ma'am. Can you open the PPT file once and put it in full screen?
Yes, sir. Please show this.
Yeah, can you hear me now?
Yes, we can hear you. Can you try sharing one?
I'll just try once. It's not sharing, I don't know for some reason, because the shared tab is disabled.
Ask her to rejoin.
Rejoin.
Okay, I'll just rejoin.
Office.
Yes.
She has joined.
So we can see your screen, ma'am.
These hours.
Yes. No, this is ma'am is the screen. So she can open the PPT now. Ma'am, can you hear us?
You can see it
Yes.
now, right, in full
Yes, yes,
screen?
Yes, we can see it, ma'am.
Okay.
Please go ahead.
Okay, so good afternoon. So I think since 2 days or since morning also, you'll be listening a lot about scientific discoveries and more, hardcore research. So I'll be telling something very different because I've been asked to speak about, how to create a venture, startup, and then what are the roadblocks. So I just decided to not to scare you, but some of you maybe more, whatever, I don't know how many participants are there, but some of you who are very keen to, whatever your research is, whatever your ideas are, and if you really want to uh, put that into a product format and commercialize it. So this is what we do at our incubator. So I am heading one of the bio incubators that is there in IIT Guwahati. It's called BioNest. So what we do is we actually sit in a our incubator is kind of a bridging entity, which bridges academia with industry. So we help startups, entrepreneurs who are at very early stage of their startup journey, you can say, or entrepreneurial journey. And we help them in developing these products within IT campus. We have a full-fledged laboratory. And then we put that into a product where you can commercialize your product and then, of course, make money. And the vision is that whatever your innovative ideas are, it should reach to the people for which you have imagined that, if you thought of building something and you're addressing a pain point of a society, right? So you need to have that product reach to the right people. So this is where you go on an entrepreneurial path, that you develop your idea into a product and then commercialize it, right? So I'll be telling you more about how you build a venture and what are the different things involved here, and what are the funding things, different areas or different government agencies from where you can get the initial fund, and then go along with that. Just now my just these slides are not changing from here then now. Just give me a second.
We are checking it.
Can you, I don't know, the slides are not changing here. Can you upload from your side? So because somehow this mouse of mine is not working.
Yes, ma'am. Really.
Okay.
Ma'am, is it all right?
Yeah. Can you go on the second slide? Yeah. So, like I said in the beginning, few of you who are in the audience, they might be thinking, or let's say that you are having an idea, which you think that you can convert that into a product, or maybe you are the last year of your degree, and then you're thinking what to do next, right? So when I started, building the company, I was around. I have done all my postdocs, and I was working actually in a laboratory. And then I thought that I have this idea, and then it came to my mind. Okay, fine. If I convert that into a product, what I have to do next, right? Can you move the next slide? So the time when I started, way back in 2017, I thought of go on an entrepreneurial path. At that time, there were no, I wouldn't say no, but the startup word was not trending at that time, right? People weren't watching. There was no Shark Tank in India at that time. So nowadays you have a lot of this buzz going on, and these words are trending. So I believe that most of you in the audience, you might know these brands, right? By the way, these are all Indian brands, which are unicorns now. So, and then a lot of you also watch Shark Tank. And so these things are, very trending these days. Can you move the next slide? So when we think about entrepreneurship, we always think about this, right? That you have a fancy office, corner office, and then you have a fancy car, you're living a luxurious life. So this is what when you see all those successful entrepreneurs or successful business people, this all comes to your mind. Ki bhai entrepreneurship yahi hai, okay? Can you move to the next slide? But in reality, it's a hard path, and like I think anything else in life. So reality is this: you really have to struggle. It's a very difficult journey. Again, I'm not going to scare you. It's a difficult journey, but that's the fun part. Because when you achieve a summit, you just realize that even though the coming there, reaching the summit was so difficult, full of hurdles, but then view from a summit is always beautiful. So this is what entrepreneurship is all about. Can you move to the next slide? So let's talk about what innovation is. When you what's the difference between entrepreneurship and a regular business, right? So business is something where you buy something from, say, from the wholesaler, and you're selling it. In a way, you're not making it. You're not doing the innovation part. You are not imagining the product. You're just buying it from somewhere and selling it like a retail shop, right? So that is what business means. But when you talk about entrepreneurship, the word that you cannot separate from this word is the innovation, right? So by innovation, and again, there are two words which are generally used interchangeably, which is innovation and invention, right? So invention is something that you create something new that was not there in the world. You create something new, but invention can not always be for society. It could be a thing which you cannot always monetize, which you cannot always say that this invention is going to solve a major problem of our society and I can monetize on that. So this is the difference. If you have done an invention, but again, if your invention can reach the society and again you can monetize over it, then it becomes innovation, right? Can you move the next slide? So let's see, let's come to this point that, okay, fine, you have an idea, right? And so you have to ask these three questions from yourself before you move on to this different journey other than from academia. So Fine, let's say you have an idea. So you say, fine, you have got an idea, but bhai, next kya hai? So you have to ask second question, ki, is my idea going to bring any change in the society? ठीक है, आइडिया तो बहुत लोग सोच लेते हैं, I have to go there and I am going to make this, I'm going to make this app or anything. But is your idea or is your innovation is going to bring any change in the society, the way society works, the way society operates? If the question to that, if the answer to that question is yes, then you ask a second question: Ki, will my idea will be acceptable to someone, or should my idea be having takers? So by takers, I mean both the people. The takers mean who's going to put money in your idea, right, to develop that into a product, or ultimately when you have done the you have made the product, ultimately will the people are going to buy your idea, buy your product. So there are two takers. So there's a second question you ask from yourself, ki, okay, fine, I know my idea could bring change, but whether this idea will have any takers, right? If again your answer is yes, then you ask a third question, which is important when you talk about business, and when you talk about that is totally a business part. The third question, that whether your idea is a profitable idea or not, because at the end of the day, you need to make money, not just for yourself, but it's all about, it circles back, because if you don't monetize, if you don't make money, that you cannot think about new ideas, you cannot develop more products, and again you cannot reach to the people which actually you thought upon, that I will bring this product to these many people, these many segments. So these three questions, if this is yes, then you move to the second part of entrepreneurship, which is more into the implementation part. Can you move to the next slide? So okay, fine. So let's say I'll just this slide I will explain you with my example, with the idea on which we have built the company. So let's say you thought about, like I told you in the beginning, that whether your idea will bring any change. So when you think about an idea, there is an old saying that necessity is the mother of invention. So again, it comes from innovation also. When there is no necessity, why will you innovate, right? When you see a problem, then only you think about ki bhai ye problem hai, what solution I can make, right? So then what unmet need of the society which you are addressing with your idea, okay? And if you have, so that's a bullseye. If you know that this is the unmet need of the society, and now I have to think based upon my expertise, my education, my training, now I can suggest one solution to this unmet need, right? And then you have your what will be the selling. So again, like I said, at the end of the day, you have to sell that product to the people, right? So what is the key selling point of your solution? Whether it is the price point, that suppose the solution which already is there in the market, suppose that solution is very expensive, right? So then you're playing on the price point. You're making your solution much cheaper, right? Say if the MRI is costing around 5,000-6,000. So if you are making an MRI machine which is very portable, and again it is costing you say about 500 or 600 rupees, so that is a huge difference, right? And then again, so price point can be one selling point. Then other thing is that you are making everything very robust or point of care, right? So, and I'll explain you with an example in the next couple of slides. So that is what is your solution and what is the USP of your solution. Then comes, so you should never think that you are the only person jisko problem dikh rahi ho, जिसने solution bana liya, right? So people might have seen that problem, and I'm sure there are many people who might have thought about a solution. But then, so that is the people who have thought about the solution of the problem that you are also addressing will become your competitors, right? So you should know what are the other products which are there in the market. What are the other people who are thinking the same way that you are thinking? So they will become your competitors. So in business terms, you say that they are going to take your market share. Okay. And then, of course, when you buy any product, right? So you start using it. Then after some time, they say, okay, we have a better version of this product. So this is kind of a marketing strategy also, and this is kind of with changing technology. So your solution should have all these qualities, that it should have enough room so that you can upgrade, you can change, so that after a while, technology should not become obsolete, right? So that latitude you have to add in your innovation. Can you move to the next slide? So I'll just tell you with an example which will better you can understand it better. So when we started to build this product way back in 2017 or 14, let's say. So at that time, we were thinking that, UTI, urinary tract infections. So this infection is fairly common throughout the world. Every third person has UTI once in their lifetime. Okay. Then, and the diagnosis for this UTI is generally the gold standard is urine culture. So the patient gives his or her urine sample, and then that sample, few drops of that sample is put on an agar plate, which is generally clad agar or chromogenic agar. And if that person is really infected with any kind of UTI bacteria or uropathogen, after 18 or 24 hours, you get a bacterial growth on that plate. So this is the gold standard, right? And then based upon the microscopic studies, a little 2 or 3 biochemical testing, the clinician comes to know that which specific bacteria is infecting that patient. Now, here are 2 problems. So we can say when you talk about in more innovation terms, we have 2 pain points. The first pain point is the turnaround time for the results. You have to wait, the patient needs to wait at least 18 to 20 hours to get the testing done, to get the results, right? So that's a huge lot of, and this is okay when you have mild UTI, but especially the patients who are actually, in the post-operative, in the post-recovery stage after a major surgery. So their immune system is already compromised, right? So at that point of time, and they frequently catch UTI because there are a lot of catheters and all those medical devices are going inside their body. So very easily they get infected with UTI bacteria, right? So that is, and that point of time, you really need faster diagnosis. You don't, if you wait for 18 hours, that patient might die also. And doctors cannot administer antibiotics, at least to the post-operative patients. Once they know the results, then only they can administer antibiotics. So that was first pain point. The second pain point was that for doing this testing, you require a full infrastructure. You need a trained personnel, you need incubators, you need a lab, you need a laminar flow. So all these things you need, right? So these are the 2 pain points. So when we started working on this solution, we thought that we are going to, so price point we were not addressing. We were addressing these 2 pain points, that we should get faster results and our system should be point of care. Okay. So then our solution came. Can you move to the next slide? So then we designed sensors. We designed nano sensors, which you can get the results in just 15-20 minutes. And this test gave you the same information as the gold standard was giving, right? So I just explained you this. I'm not explaining the science part of it, but I'm explaining you that how we address the pain points, how we came to a solution, keeping in mind that we are addressing one disease which is so common in the entire globe, right? And we have 4 or 5 bacteria which are commonly found in infected urine samples. So this is what we were addressing. That was a solution, right? Can you do the next slide? So let's say that you have an idea, right? What are the next steps? Abhi kya karna hai? Because in the beginning, when you have an idea, you never have money. And you don't even know how to develop the product. And if you are thinking of a tech-driven product which involves a huge, technology component to it, you need a full-fledged laboratory to work on. And in the beginning, you cannot have this expensive infrastructure invested from your side, right? So these are the 3 steps. First of all, so when like maybe 10 years back, or we don't have these facilities in India, of course, where any other government agency are providing you with all kind of infrastructure. But this is a very good time. That is why this is a time to start up is now, that now you have all facilities and everything in place, where you just have to come with an idea and then develop on it, right? So the first step is that you just find an incubator. So incubator, in literal sense, uh, it's kind of it is kind of an entity which incubate the budding startups until they are ready to hatch. Okay, when I say hatch, that means they become self-sustainable, their product is developed, they have raised money, and now they don't need the incubation, right? They are on their own, right? So until that time, you should be incubated. And in incubator, what you can find, you will get the full infrastructure, and then you will get any necessary laboratory support that you need to develop a product. Say for if I take our example, we needed to develop these nano sensors. We needed to do a lot of procedures, mainly, select procedures to develop these DNA. And then we need microbiology facilities in which we need all kinds of assays for bacterial testing. So we needed all this, right? So, and this is generally provided by the incubators because they have all the infrastructure laboratories which are used by the startups in the beginning, right? And then one thing that is very much needed in the beginning is the hand holding. Not in the terms, because if you have to develop a product, which means you are doing something which you already know. You know the protocols, you know the procedures, you know the science part of it. But then you don't have or very little idea as to how to build the company. You have no idea in the beginning how to bring in money. So when I started, I didn't even know the terms like what is valuation, what is all those accounting terms. I had no idea what they mean. So this hand holding is very necessary, which is generally provided by the incubators. They handhold you in these steps, how to make a company, who to approach to make a company, and then how to make even the balance sheets of the company. In the beginning, you cannot have money to hire a full-time CA who can make your balance sheets and all those things. So all these things you need to know, and even if you are outsourcing it, you need to know that how this is happening, how the cash flow is happening in your company, how the cash is coming, how you're utilizing the cash. So all these things, what is your burn rate, meaning that how much cash you have to survive over, say, next few months. What is the runway money you have, means how many months you can survive with the money you have. So all these hand holding is very necessary, and this is what our incubator, any entity like us provides to a budding or early stage startups, right? And obviously, these incubators help you in raising fund either from government or from private equity. Then comes the second part or second step, which is very important, is to get the initial capital, right? And that you do generally by two ways. Either in the beginning when you are building a product, that means when your product is at ideation stage, or you in startup jargon, you call it that you are a ideation stage startup or a early stage startup, right? So at that point of time, investors generally, they are very reluctant to put money in you, because when investor puts money, they want it back. And they know that if you're building an idea, they are not very sure that this idea, whatever you are saying that it's going to do this, whether after 2 years or 3 years down the line, you will be coming up with the product, what you're saying. Right. So that is why they don't want to take the risk. So at that point of time, in the very beginning, either you take government grants or you put money from your own pocket, right? So that is called bootstrapping, when you put money from your own pocket, either from your family, friends, which is your money, right? And then comes the government grant. That is, an Indian government has got many. I'll be telling you what are the different funding schemes, right? So, and you can get this grant. And then the third step is that you have to hire a minimal but very efficient workforce. So in the beginning, you cannot, of course, afford to hire too many people and very good people, because if they are very qualified, they'll be needing more salary, right? So, and you, of course, in the beginning, you cannot afford that. So generally, when startups are formed, say generally 2 or 3 people come, forward and they form a startup, right? So generally, say when I made my first company, so it was like that I was talking to one of my friend and we decided, and he has got a different set of expertise, and I was having a different set of expertise. So we thought, let's build this. So in the beginning, generally founders itself, they work and develop the product. They work in the lab, like if you have a chance to visit any of the incubator center, you will see that mostly the initial product development is done by the founders. So there are two benefits of it. The first is the foremost is that, the founders, they know more about the product because they are the people who thought about the product. They are the people who actually thought of the solution and how this product will look like. So they are passion driven. So in the beginning, they are the people who are actually needed to build the product. So generally in the beginning, the first prototype is always built by the founders itself. And then, by the time you raise money from other sources or from investors, then you can hire people. When your product is ready, you have made the prototype, you have made the MVP, which is the minimum viable product, which is ready for the market. And then, of course, you need to hire more people. Can you move to the next slide? So I'll just tell you briefly what are the different government schemes that are there, by the Indian government. Can you move to the next slide? So this is the first very popular scheme, which is Biotechnology Ignition Grant or BIG grant, which is given by BIRAC, Government of India. So basically, this is a scheme which is mainly for the students or for the entrepreneurs who are building the product in the biotech sector, be it in healthcare, agriculture, food technology, clean energy, also waste management. So all these things. So then it's a very prestigious scheme by Government of India. So they generally give you 50 lakhs of initial grant, which they call it early ideation phase grant. And this grant is mainly given to build the proof of concept, right? It's a very initial grant or the first stage grant. And then again, the same type of grant is given by DST, Government of India, which is called as Nidhi. So Nidhi has three schemes. One is EIR, the other one is Prayas, and the third one is SEED. So EIR is generally for the entrepreneurs who, like students, maybe like you, who have an idea and you still don't want to form a company. Because once you form a company, there are a lot of compliances which you have to do with the government. So anyways, if you're not interested to make a company right now, but you just want to test your idea, whether it is working or not, just very, very initial proof of concept. So for that, this is a very good grant, which is in this case, you don't have to make a company, but you also get a fellowship per month and a little bit of a kickstart grant. This you can do in your current affiliated institutions also, which is called as EIR, which is Entrepreneurship in Residence. Then comes the second level of grant, which is called as Prayas. So Prayas, in that, once say you are going this way, that you have taken the EIR grant and you were taking fellowships and you were got a little kickstart grant of 3-4 lakhs rupees, and then you build you're successful in completing the proof of concept, right? And now you said, okay, fine, now I know that this product is going to work, and I can with few iterations, with few tweaking around, I can make it make this a product into a profitable product and market ready product. Then you form a company, you incorporate your company anywhere you like. And then you take this second level of grant, which is called as Prayas, which is generally always given to a company. I think they give around 10 lakhs of, that's just the highest amount they give, which again helps you in building the prototype, right? And then the seed grant comes from DST Nidhi. This is given in the form of investment. So the ceiling amount could be 1 CR also. 1 crore, and in this case, depending upon which institution is giving you this grant, it is not a grant; it's basically investment. So depending upon whatever negotiation you have, so they might take some equity sharing in your company, or they can give you a debt of 1 CR, which you have to return back. Ultimately, this is for those products which have already passed the proof of concept, the prototyping stage, and you are making it market ready. Okay. And then again, there is Atal Innovation Mission. They have very exciting schemes for early stage startups and from MSMEs, SMEs also. And there's another one from STPI, Software Technology Park, Government of India, which is called as Chunauti. So Chunauti is again just basically they give you grant up to 25 lakhs. It's not a grant, basically it's an investment. And then again, STPI gives you, if you have hired 2 or 3 interns, so they also give some kind of intern amount, monthly stipend to 2 or 3 of the interns that you are hiring, right? So these are the 5 main I've listed here. There are many, but these are the 5 main ones which are generally utilized. And this is again very exciting grant. I just missed that Startup India Seed Fund scheme. So this scheme, again, they give you amount around 20 lakhs is given as a grant. And if you want more, they can give you up to 1 CR also, but then it is in the form of debt or investments, right? Can you move to the next slide? So these are the 4 levels, right? If of a product journey, you can say. You start with ideation, you go to proof of concept, and then you build the prototype, and then you scale up that. You do validation with, say, in the case of any diagnostic kit, that validation is done with the patients. And once you have validated it, then you put that product into the market, right? So this is how a product or an idea takes off from ideation stage to the market entry phase, right? Can you move to the next slide? So these are 3 takeaways. Next slide, please. So first of all, the first takeaway is that you have to define your business model. Now, I understand when you start your venture and when you start on this entrepreneurial journey, you don't really understand what business model is. At least I didn't know. So at that, but still you should keep in mind, you should have a little idea about how you are going to sell your product, how you're going to set up the business once you start building it. So either you can, so one way is, to define your model is that you can make the product and you can sell the product also all by yourself, right? So that is one model. Second is that you can make the product, get the IP, and then you can license it to some other company, right? Who is going to market your product. Many people do that, right? So this is called as licensing mode. Okay. And another way is that if you already have you have built a product, you have built a suppose technology, or maybe someone else has made this technology, you can transfer that technology to your company's name, and then you can start building it for commercialization purposes, right? Can you move the next slide? So the second takeaway is that you should know your ecosystem, right? Who are your competitors? Who are the people who are putting the similar product in the market? Who will be competing with you in terms of market share? And you have to, always observe them, or I don't want to use the word stalking, but of course you have to see what they are doing, the way they are selling the product, what is their marketing strategy. So all these things you should know that who are, because they are the people who will be taking your profit share if you have in plain terms. So you should know, and then that also helps you to understand your product better, and and your market better, your customers better, right? So that you should know your ecosystem. That is not just the competitors, but also customers. What your customer actually needs. Say, for example, if you have put one version of your product in the market, the customers say that, it's very difficult to hold it. It'd be easy if the design is like that. So all these you should know, right? And what other people are providing, they say, okay, there's a similar product in the market which that XYZ company is providing, but it is very beautiful looking. It's very light. So all these things you should know your ecosystem. That will help you in actually, building your company and your product both. Next slide, please. So then comes the third takeaway, which is fine tuning your entrepreneurial skills, right? And this is very important, and you have to do it all the way long. How to make a good pitch. Sometimes, when even the incubators that I have in BioNest, when I see their pitches, because we help them curate their pitches over, say, so there is one of the incubator, I was looking at his pitch that he gave me 2 years back. And now what he does, it's a huge difference, the way he has evolved. So that you have to fine tune all the time. And then you have to meet people who are into the same ecosystem. You need to make, because in the startup world, if you're entering, networking is very important, very important. Not just networking, you have to build your personal brand and the brand, what you're making, your product brand. So all these are 2 different things, but you have to do them together. So it's very important because people should know you. if they know you, then they will know what you're doing. So, and that will actually, that's a way to endorse your own product, right? So can we move to the next slide? So I'll just tell you very briefly in like next 2-3 slides and I'll finish that what we are actually doing in Bionest here. And like I said in the beginning, we're actually bridging between academia and industry, and we are helping startups to, take their product from the lab bench to the market. So next slide, please. So we provide everything. You can just, if you have just an idea, you can come to us. We'll help you build the company. I mean build a venture. I mean all the paperwork that is needed to build the company, and then we'll help you. We'll incubate you with us. We also have funding, a lot of startup funding with us, which we give to the startups for the initial POC and prototype development. And then after you have done this, we help you take off, means that we'll help you getting more equity investments and the successful graduation. Next slide, please. So we can skip this. We can go to the next slide. So these are the prime focus that we have right now. We are incubating companies in these thrust areas. In, like I said, it's a Bionest. So we are mostly supporting companies who are into the biotech domain. So in healthcare, agriculture, food tech, we have a lot of companies who are mostly in the agri-tech sector, because Northeast has got many startup ideas which are based on agriculture technology. So these are the thrust areas. Next slide, please. So we have two kinds of incubation models. One is the physical incubation, in which you come, you work here, you use the laboratories. So we have office space for you, laboratory space. All the physical infrastructure that we have is accessible to the startups, and they use it. Then comes the virtual incubation, which is generally they cannot use the physical infrastructure, but we connect them to all the right stakeholders, be it a ecosystem connect or investor or the intended market, right? So all this is done virtually with the virtual incubators. Next slide, please. So what you will gain, once you come with to us, to IIT Guwahati Bionest. Like I said, all these things, mentoring, funding, product development, collaborations with, so like many of our incubators, when they stuck at some point, we help them collaboration with IIT Guwahati faculty, where they can, according to the expertise, they get the solutions to whatever problems they're facing in product development, which you always face anyway. Next slide, please. So these are a few facilities that we provide. We have a high-end laboratory. We have modular office spaces and collaboration opportunities, whole mentoring. We have a full set of mentors which we handpick from all over India, which keep on mentoring the startups from time to time whenever it is required. So we also provide IP and patents. We facilitate them, the IP filing and all those filings. And of course, we help your marketing. So if you are building a product in healthcare space, regulatory compliances are a very important integral part. Before that, you cannot put your product into the market. So we help in all these certification, government approvals, who to connect, which CRO to be approached for doing all those things, right? Next slide, please. Yeah, we can skip this slide. Can we go next slide? Yeah, next slide, please. So this is how we do. We scout ideas that we do all year round. We keep on scouting good ideas through many open programs also, like one we are hosting in the month of April. So this is Ideathon, where students can participate and they can have their ideas on any of the problems. Doesn't matter which sector it is, but if you have, you just have to state your problem and just say what ideas you put forth. And then, so this is what we call it scouting. And then if we think that your idea has got really good potential for market, we can facilitate some of the fundings for you. Either it could be form of an EIR, we facilitate that, and then, or maybe direct funding from our incubator. And likewise and then we help you build the product, and then all the regulatory that is done, and then your market entry. Next slide, please. So these are the two funding schemes that we have at present with Bioness. One is from Northeast Council. So basically, we provide funding up to 10 lakhs to per startup, but the startup should be registered in any of the 8 northeastern states. That is one condition for this funding scheme. And the second condition is that the startup should have done the POC. Like you have done the proof of concept and you are building a prototype. The second scheme that we have is from Startup India, which they call it Startup India Seed Fund. Again, this is given to a startup company which is within 2 years of its incorporation. And this is not Northeast specific, pan India. Anybody from entire any of the Indian states, they can apply for this funding with us. So this is grant support of up to 20 lakhs, but if you need more than 20 lakhs, then that is given in the form of investment or debt. Yeah, next slide, please. So again, so this is the initial funding that I talked about, but when you are done with the initial funding or common grants and you want to, build the product, build the market strategy or marketing strategy of your product, then you obviously need investors. So we have these ecosystem partners, or these one of the India's number one, VCs right now, like Social Alpha, Marwari Catalyst. So we have MOUs with all these VCs, and they periodically they keep on talking to our incubators. They most of them, they do know what kind of incubators we have, what kind of companies we have, and recently 2 or 3 of our companies, they have been taken over by Marwadi and Head Start. They are interested to invest in those companies. So this is kind of an ongoing exercise that we do here. This we generally once we raise funds for them from these investors, the companies, they generally exits because now they are on their own. Next slide, please. So this is my team. Professor Dipankar is he's the PI, principal investigator for Bionest, and this is me. And we have a very efficient team, small team but very good team. Next slide, please. Yeah, so that's it, I guess. I think next slide is the last slide. Yeah. Yeah. So if you have any questions, queries, please go ahead.
Thank you, ma'am, for your valuable talk. We'll now begin the question and answer session. Now, there are a few questions from the participant. I'll just take one question. The next questions you can address in the chat box. The question is, sorry, MTech student can propose for grant for the project they are working on. I think an issue student face while working is because project completion would be limited by tenure parent institute or lab.
Just can you repeat? Just a moment. I can just open the chat box.
Yes, ma'am. You can take the question from the chat box with the last question in the chat box.
Yeah, just access the chat box maybe. So.
I mean, it's asking that if a MTech student can propose for a grant for a project.
Let me just access the chat. Can you go on? MTech student, okay. Shall a MTech student can propose a grant for the project they're working on? See, as a student, if you want to propose a grant, if you are working on a project in your, say, an institute while doing MTech, so whatever project you are doing, you will be developing an IP, right? Your patent. So that IP will be owned by your institution, okay? And after that, if you say you have done your MTech and if you want to open a company and if you want to develop their products, you have to transfer that IP to your company. Again, while doing your MTech, say for example, if you want to apply for any of these startup grants, you can do. But again, the only issue occurs here is that first of all, if the grant is given to you, okay, you cannot avail the grant until you make a company. That's one thing. And if you do make a company and if you get a grant, then you have to transfer the IP or technology to your company. Because say you are doing it, say in IIT Guwahati, right? So you have developed that product in IIT Guwahati, IP will be owned by IIT Guwahati, even though you are one of the inventors. And if you are interested to develop that product now in the form of a commercial product, then of course you need a company. So you can open a startup, but then again you need to transfer that technology to your company, because that is a legal way to do it. Okay.
There's one more question, just as posted. I am from IIT Guwahati itself. Actually, I just wanted to know if we want to approach biomass for incubation, should it be through the online application given in the website?
Yes. So if you are from IIT Guwahati or from anywhere for that matter, you can just go on the Bionas website. You just click on apply for incubation, right? There's a little Google form if you already have a company, because generally we incubate companies, not individuals, right? So if you already have a company in place, you just click on that button, which is apply for incubation. There's a Google form. You fill that form and we will receive that all your responses, and my team will connect with you. So generally, you just have to give a little presentation that what your product is, what you want to develop with Bionest and all these things, and then you are through for incubation.
And the other question is, can BTech students also can enroll for these initiatives or not?
Like I said, you can, but you need to have a company. Right. That's a must, because we don't have a company in place actually, but we just started the process of getting. Yeah, I mean, fine. Once you are done with an incorporation, company incorporation takes place just 1 month, right? Once you have done, if you are in IIT Guwahati, please come meet us. I'm always here. Right, you can meet me and let's have a chat that what you are building, and then we can help you. We can tell you the steps. And if you are getting in the process of incorporate, it will just take 1 month, right? And after that, you can apply for incubation. But for meeting me and seeing the facility, you can come anytime.
There's one more question from Alok. Can you just take that question?
Yeah, I'll just see. Alok, innovated. Need a partner. So Alok, I think so. What I need to ask you actually that how you already have a company, or you I mean planning to, how you're this answer comes from your response, that what are you visualizing your product as? Do you have a company right now? Okay, you're a PhD student, fine. So see, there are two ways you can proceed. Either see, if you want to commercialize a product, there are two ways. Either you do it yourself. By yourself, I mean that you have to incorporate your own company and then commercialize it. Second way is that if you don't want to do it, say, I have seen most of the PhD students, they want to go for postdocs and pursue their career in West. So that is fine perfectly, but then you can transfer this technology if you have already got the patent. If you don't have a patent, please apply for the patent. Once you have the patent, you can license this technology to any of the company. We can facilitate that, because we do this matchmaking, the right technologies to the right founders. If you are not able to do, if you don't want to do it by yourself, we can match your technology to the right company, to the right founders. They will commercialize it. So in a way, you are licensing your patent to them. What you are going to get the royalty once this product comes to the market, right? But if you want to build this product on your own, then it's that you just file a patent, get a patent, and then you can incorporate a company, and then you can do it by yourself.
Thank you, ma'am. Ma'am, rest of the questions, please write them in the chat box.
Okay, yeah. Can you go up, Kanmuna? I just want to see the other questions.
Yeah, just one other question there. One question is there. They wanted
research patterns. I'll like to listen about the research patterns and commercialization, please. Yeah, that's a very good question, Prashant. See, this is what we are doing, and we are actually almost in the final phase of drafting this program, which we are calling it, tech transfer accelerator. In this, what we are planning is that, and we are doing it with IIM Ahmedabad. See, because IIM Ahmedabad is that entity which brings a different set of expertise in the business expertise, and we are the people who are technology builders, right? So this is the marriage of these two institutions. Two thoughts, you can say. And this is what we are trying to do. Say, many of the academic institutions like IITs, they develop technologies, right? And most of these technologies, they don't come out into the market. And this is the pain point which, as a Bioness, as an incubator, we are addressing. So what we are doing in this way, if you have our technology, what we will do, you just tell what the technology is and what pain point you're addressing, and we will find the right company partner for you, right? If you don't want to do it yourself, we are going to find the right company partner for you. In the sense, it could be a startup, it could be established conglomerate also, which will only develop your patent or your technology into a commercially viable format and reach to the right people which you have designed it. So we can talk it over. Please be in touch with me, and definitely, because this is what we are doing and we are planning to do, and then we are also planning to fund this entire thing, the commercialization process. It is in the structure phase, but please be in touch, and hopefully in the next 4 or 5 months we'll make this happen, the translation or the technology commercialization part.
Thank you, ma'am. Thank you for the talk and answering all the questions. Thank you, ma'am.
Thank you. Bye.
With this, we extend our gratitude to everyone for their valuable time and a successful morning session. There is a lunch break now. The next and the final session will commence at 2 p.m. Participants are requested to be present on time. Quiz will be held after the afternoon session. Thank you all.
Pro.
Pankaj sir, can you hear me?
Yeah, I can hear you here.
Okay, so we'll start in 10 minutes.
Yeah, sure.
Okay, okay.